Hello? Can everyone hear me? Hello? All right, let's get a thumbs up in the chat if everything is working good. Yay. Okay, good. I'm glad everything's working. Okay, well, welcome everybody to the first Whose Gene Is It Anyway stream. I think this is going to be a lot of fun. The basic concept is going to be pretty straightforward. Today is going to be a little bit different, just because it's the first one. I want to kind of set the tone. But moving forward, the way this is going to work is I'm going to pull a ridiculous idea out of a hat, and then we're going to go through the process of designing the DNA that would accomplish that ridiculous idea. I have a very long list of these kind of crazy, ridiculous ideas. Some of them are a lot of fun. Some of them are going to be a very serious challenge. Other than today, I'm not going to have researched any of them in advance, and so basically, the idea is that I'm going to have to research and design and do everything live. I'm going to probably ask for some of your help, because, if it's just kind of me reading through papers, it's not going to be nearly as exciting. But as these things go, I think this should be really fun. So we're going to get going in just a second. I'm just kind of making sure that everything is looking good. Okay. So I believe we just need to adjust the stream settings really quick. So I'm gonna just see if I can sort that out because I don't know why. This is also my first time using Streamlabs, so we're kind of learning on the fly. So, we'll do with it what we can. Okay. So what is going on? Why does this look like pork? Color, default color range. This is funny. I thought I had fixed this beforehand, but I guess not. Okay, whatever. This is fine. So you know what? If this stream isn't perfect, that's fine. It'll get better as we do this again. Like I said, learning on the fly. Okay, I can turn the audio up a little bit. Is that better? There were a couple of comments that it was a little bit quiet. So is that better? Can everybody hear me good? Because if it's too quiet, I can turn it up a little more. Okay, cool. Screw it. We're going to get going. Okie dokie. So, with that as it is, so the idea for today is kind of fun. So to give a little bit of background, there's two companies that have done something really cool. I don't know why it keeps doing that. But yeah, so the companies have done something really cool. So these are the two companies here. So the first one is Perfect Day. They also go by Moofree. I don't know if they've changed their name or what the deal is. And then the other one is Real Vegan Cheese. So the idea is pretty straightforward. What they've done is they've taken yeast and they've genetically modified it so that they produce the proteins from milk, so that they can essentially grow or brew milk, which I think is really cool. And so then, the idea is for something like Moofree, they're going for like that range of products. So, ice cream, cream cheese, that sort of thing. So what they do is, they grow the proteins and then they mix them, extract them, and then they mix them with plant-based fats to kind of stand in for the usual milk fats, and then sweeteners and flavorings, all coming from plants, so that you can get essentially a vegan cheese or vegan milk, which is kind of interesting. But like it'll taste like milk, it'll look like milk, it'll do the thing like milk, which I think is pretty neat. Real vegan cheese, same basic idea. They modify yeast, they get it to make the proteins, they make their milk stuff, and then they turn it into cheese. So I was thinking about this, and I was like, this sounds really cool. And so I pulled up the patent. Now, I don't like patents. I think patents are shit. Frankly, if we had no patent system, that would be wonderful. The idea that you can own an idea is ridiculous. That being said, I take great pleasure in finding gigantic loopholes in patents and then breaking them, because if you can do that, you can then open source the idea and then nobody's allowed to sit on it, which is fun. Not saying necessarily that that's what's going to happen today. I'm just saying that this is a great baseline. However, especially with patents, if you're going to be a dick about it, I am, even more inclined to fuck with you, because it's how I do. So the basic idea is what I've described, right? They make the proteins, blah, blah, blah. And so in the patent, they detail all of the different details. Ooh, wow. Well, thank you. I can't pronounce Prithvi Quantum. Thank you very much. Appreciate that. But yeah, so in their patent, I'm reading through it going, okay, how does this work? Because I'm just curious. Like at first this started as like, this is a cool idea. I would like to know how it works. And then I hit the, come on, what happened? Control F. Why? What the hell? Why you do this? Cat. You know what? My laptop is being a little glitchy, and it's kind of annoying. Come on. There we go. Okay. This is what I was looking for. So this right here, hopefully all of you, come on, highlight, please. So yeah, this is hopefully what all of y'all can see. It is the most obnoxious list. Here, I'll see if I can make this a little bigger. Oh, too big. I broke it. Where did it go? Oh, here we go. So yeah, this is their list of all of the animals that they claim ownership of. So basically, you're not allowed to touch any of the proteins that come out of these animals. And they really tried to be inclusive. So, you've got the usual ones like cow, sheep, goat. But then they really leaned into the, just being the worst. And so, you've got orangutan, gibbon, mandrill, pig, fox, fucking woolly mammoth. Like, they really thought that they were like, yeah, no, we're going to be inclusive. Like, might cut out. No, everything should be fine. So, yeah. In theory, you're not supposed to be allowed to patent nature, which in theory is what they're doing. So you could probably take them to court and try and fight it that way. However, I don't fuck with lawyers if I can avoid it, because, yeah. So I like the more clever option, which is, well, okay, if they're going to be dicks about making this ridiculous list of animals, what if we just found one that wasn't on the list? And so some of the astute viewers may have noticed that there's a few things missing on this list. I mean, there's a lot of mammals, right? Like, obviously they're not going to do it. Well, I'd say obviously they included woolly mammoth. I don't think obvious. We've passed the point of obvious at this point. But they're really leaning into just being dicks about this. Except they forgot a really obvious thing on this list, and I have checked this. Like, they've got echidna, they've got like a possum, like they've got things that, let's be honest, you would not really want to make milk out of. But they've included on this list just because they're the worst. They missed deer, like normal white-tailed deer. which I checked recently has been patented. So, so what we're going to do today is we're going to find all the proteins that are this patent lists as needed for making milk, and we're going to make a piece of DNA that is going to produce all of them from deer, the one animal on this list they didn't include. It also is not in the real vegan cheese patent either. They only do, I think, cow and goat. So yeah, we're gonna make some deer milk. And so I was kind of thinking about this. The reason I even was starting to think about this is because, to the surprise of hopefully nobody, we're in a pandemic. So, there have been moments when, the stores are empty, with some notable exceptions. Around here, at least, things that have not run out include sugar. And I don't think sugar would ever really run out just because of the ridiculous scale at which it's produced. Like, it's basically a waste product because it's so subsidized. So yeah, we're basically gonna. So I was like, yeah, okay. What would be great is if we had these yeast. Obviously they're not going to share theirs, but it's like, if you could make your own, if you had your own yeast, when things are running low, say you want to bake some bread, you've managed to. Ooh, why? Thank you, Merlin. Say you wanted to, make some bread or whatever, or just, enjoy milk products like. I like me a sandwich with some cheese on it. But if you can't buy cheese because everything's sold out because everybody's stockpiling it like a nutcase, then, it becomes a little more difficult to get things. But if you could brew it, like I grow huge amounts of yeast for all kinds of things. Like I do a lot of brewing. I do a lot of baking. I'm actually, for those curious, I'm actually working on a video basically titled "There's No Such Thing as a Yeast Shortage," because for those of you who have decided to, like myself, decided to, start baking to deal with the fact that all the stores are sold out, or just, to reduce the number of times you need to go out, like a responsible adult, you may have noticed that, things like yeast have all of a sudden become very difficult to come by. Ooh, we had another donation. Well, thank you very much. So, considering that things like yeast all of a sudden have become very, very difficult to come by in the usual, packet form, I'm doing a video where, all the yeast biologists are sitting there going, there's no such thing as a yeast shortage. It doesn't exist. Because there's no such thing as a yeast shortage. Yeast is everywhere. So I'm making a little video, showing this. But anyways, point being, I have loads of yeast. I have loads of ability to make things with yeast. So if I could grow milk, so that I could bake with it or make cheese or whatever else. That sounds great. That sounds like a thing that everybody should have. In the same way that everybody's brewing their own beer, I think it would be awesome if you could brew your own milk. And then I was thinking about this. I'm like, okay, but then one of the other limiting things, because I like egg bread. My favorite kind of bread to bake has eggs in it. And I don't fuck with vegan eggs. None of this soak some flax in water to get weird flax goo. No. Not about it. Not into it. I refuse. But most of an egg is just protein, right? So in the same way as if you can just make milk yeast, what if you could make egg yeast? So basically the idea is I want to see, and hopefully this stream doesn't take too long. Hopefully we can get this done pretty quick. But ideally, I would like to walk out of the end of this stream with 2 plasmids. One that makes all the proteins for milk, and one that makes some of the proteins for eggs. I already did a little bit of looking into this. And, egg is a little bit chunky. The proteins are utterly massive. So I'm also trying to, for this particular stream, I'm really trying to lean into making sure that the thing we make would actually be synthesizable. This is something that you could submit to a DNA synthesis company and they could make it, and then just all you got to do is put it into some yeast and you're good to go. So that would be ideal. For future streams, I'm not gonna not necessarily gonna hold myself to that standard, especially if the idea. Ooh, thank you, Ignacio. Oh, that's awesome. I'm glad I got you interested in micro. Micro is dope. But yeah, so for this one, I want to make sure that the plasmid is actually, you could actually make it. But for future streams, if the idea is truly ridiculous, then I'm not going to hold myself to that. It's just going to be like I'm going to make it just for the sake of making it. And then one thing that I'm going to do that I think is really important is at the end of every one of these streams, I'm going to post. I'm just going to, on GitHub, I'm just going to like make a folder for all of the plasmids that we end up making on these streams, so that anybody who wants them is free to play with them. My one request is that if anybody feels like getting them synthesized before I do, please share. But other than that, I think just open sourcing all this is going to be a lot of fun. And one other thing that I've been thinking about, and this is sort of the last point I'm going to touch on just before we actually like get into the work, is that especially as these streams go on, like there's going to be certain ideas which are viable. And so I'm not sure if I'm going to do a vote or if I'm just going to kind of like put forward the one I think is like the most likely to work. But basically be like, okay, well, we've designed the DNA now. But obviously getting DNA synthesized is pricey. It can be expensive. Like depending on how complicated the thing is, it can cost, well over $1,000. Or it can cost 200. It kind of depends what you're doing. But, doing, maybe a little fundraiser while we're doing this could be kind of cool, because then we can kind of all pitch in, I'll get the thing made, and I'll get it into yeast or whatever, and then I'll just distribute it to whoever wants it. Like I'm, at basically cost of making making stabs to distribute and shipping, which that seems fair to me. And that way, everybody gets to play with some cool DNA stuff and play with some cool organisms. Everybody wins. This sounds great to me. So hopefully this is something that you guys are into as well. But yeah. So with that out of the way, I think we have basically covered all of the intro stuff. And so this stream is going to be pretty chill. Designing DNA is occasionally stressful, but mostly pretty chill. So considering that, this is not going to be we're not going to be in a rush. Today is really a very lighthearted project, I find. Ooh, thank you. This is from Omega. Well, thank you very much. So yeah, today's going to be a pretty lighthearted project. I know there's going to be about a bazillion requests for coronavirus stuff, and we will talk about coronavirus. I'm working on a coronavirus video. I have been working on it for months, but as you might imagine, supply lines dried up really quickly when the world went to shit. So I got a little delayed, but I am working on it. Things are progressing. We will talk about coronavirus, just not fucking today, because I don't know about all y'all. I'm pretty burnt out on coronavirus. It's kind of a shit show. I don't want to talk about it. Cool. Glad we got that out of the way. Okie dokie. So, have a little sip of tea. Okay. Let's get working. So, really the whole point of this is to show the research process and how the designing of things works. So this is the tool that I love when it comes to designing DNA. There are many. SnapGene is one that a lot of people really like. I'm not a SnapGene fan personally. It's I don't know, it's fine, but not into it. This is called Genome Compiler. So this is a really, really awesome program. So I'm just going to set this to single project view just so it's a little clearer what's going on. And now, admittedly, looking at this, if you're not a bio person, this probably looks like Chinese or code or whatever, because it is code. I mean, it's code for biology. It looks a little weird. It can look a little cumbersome, but we're going to go through it. We're going to make sure that hopefully everybody's on the same page. Okie dokie. So, to make our deer milk, because we're gonna start with. Actually, I'm not sure. I don't know if I want to start with the egg or the milk, because the egg is going to be the easier one. Because when I say egg, I mean egg white. Can't do egg yolk. Egg yolk is too hard. So for the sake of walking away with a thing that's actually doable, we're going to do egg white. But so anyways, so this thing here on the right, this big circle, this is called a plasmid. It's sort of like this is sort of like the entire compiled program, oh, that's awesome, Omega. Glad I can inspire. And thank you. But anyway, so this is like the entire compiled program. This is what's called a backbone, and basically this is sort of like most of the program comes pre-written, and all you have to do is put in the little, like the one or two things that you actually want. So in this case, oh, my mic is shrinking. And I don't know why. Okay, there we go. It's stable again. Sorry about that. Anyways, yeah. So this is sort of like the, most of the program's written. All you got to do is put in the thing that you want it to make, and this is, let's cut off, zoom out. Let me just have a look. See. Oh, that's strange. That I see what you mean by it's cut off. Allow me to fix that really quick. See, do. Oh, okay. Oh, wait. I think I can just fix this way. Sorry about that. Let me just give this a little fixie fix. That's really obnoxious. Sorry about that. Come on. Yay. Okay, that should fix it. Okay. Like I said, first time using Streamlabs. It is a learning process. We'll get there. Okay, is that better? Well, I guess there's a little bit of lag, so it's going to take you a second to see the change. But I'm back in Genome Compiler. Hopefully everything is viewable now. Okay, come on, lag. Any day now. No, it still looks like crap. Huh. Weird. Alright, sorry. Give me a second to fix this. Oh. Gotta hit the transition button. Okay, sorry. Like I said, I still suck at Streamlabs. We're learning. It's my first time. Be gentle with me. Okay. I think I've actually fixed it. I'll keep an eye on the chat, and I'm gonna just keep going. Did it work? Yeah. Okay. I'm seeing the yay. Okay, good. I can see it on the stream now. Isn't streaming exciting? This is why I record things. I can fix it in post. I can't fix it live. Okay. Anyways, back to what we were doing. So anyway, so this is what's called an empty backbone. Basically, it's got as much of the stuff in it as you could really want, so that you've got to do the minimum amount of work to just get it working. Now I've got a couple different ones open. I'm not sure if this is the one that I'm going to use. This is sort of just the one that I pulled up. Also, for the 1,012 people who are asking, yes, this is being recorded. I will leave the stream up. I understand that it's not at higher definition. I don't know how to fix that. Yeah, so that'll be fixed for the next stream. I apologize. Anyway, so yeah, this is empty backbone. Not sure if I'm going to use this one. This is just one that I pulled up because I wanted something to just show, and we can kind of think this through. So before we really talk about the backbone, let's figure out what proteins exactly we want to make. Yeah, okay. So the nice thing with this patent is, even though they are dicks, they did us the favor of listing off which proteins are actually important, because they've already done that work. Saves us some time. So from my understanding, they've got milk, and I've already looked into this a little bit. Milk contains a whole bunch of different proteins, and that's kind of what makes it, milk. So there's a few though that really make it stand out, um, as like have that very distinct flavor and texture and all that kind of stuff. Um, in terms of proteins, um, there's basically like the main ones that go into something like cheese. The are casein. So there's a few different caseins, and you can see this in the patent. So it actually specifies which caseins. Um, but that said, I mean, there's a lot of stuff in milk. Like it's these are the sort of main components. But like if you were to try and perfectly replicate milk, you'd need a whole bunch of different proteins. But that's a pain in the ass, and you can get a good enough approximation with just 3 or 4 of them. Um, so yeah, basically the ones that I personally think are important are going to be the beta casein, the kappa casein, um, and then there's another one. Oh, hey, thank you, Ricardo. Um, and then there's another one, alpha lactobumin, I believe, which I spotted it in this patent a little earlier. But anyways, so I think if we do those 3, I think that's a really good starting point. So those are going to be our milk proteins. And then for the egg thing, um, yeah, so for egg, the main component of egg white, about more than 50% is a protein called ovalbumin. The rest of it is mostly water. So if you can produce ovalbumin, you've basically made egg white. Like the more it expresses, the better. But like you're basically making egg white at that point. So like I said, can't do egg yolk. The proteins are too big. And so we're going to do ovalbumin. So the site that we're going to get these proteins from, for the most part, is called Uniprot. So you can see it right here. So I'm just going to the Uniprot link. So this is the ovalbumin from chicken. Now, I've been kind of debating this because I have a friend who is allergic to eggs. I actually have a couple of friends who are allergic to eggs. And most of the time when people are allergic to eggs, they're allergic to ovalbumin, specifically the chicken ovalbumin. There's lots of cases where if you use a different ovalbumin, so like if they're cooking with like duck eggs, for example, they won't have that reaction. So I'm kind of contemplating, like, if we're just gonna do this anyway, like, why don't we just use a different bird? Don't be boring. Don't use chicken. Use duck. Duck is better anyway. But I don't know. Haven't decided. Anyway, so the Uniprot has lots and lots of information. Frankly, more than we need. The important bit, if we scroll all the way down, is this right here. So this is the actual amino acid sequence of, in this case, ovalbumin. Yeah, it's also called albumin. I mean, the thing with biology is, there's a million names for everything, and people will call, like, there's even species that have like 4 or 5 different names because they were independently found or whatever else. It's kind of a pain in the ass, frankly, to just keep track of. But anyways, so ovalbumin, albumin, it's all kind of the same. But anyway, so this is the amino acid sequence. This is the thing that if we get yeast to make this, we're like most of the way there. So as you can see, if I were to just try and highlight this, like it highlights a bunch of other crap, which is not helpful. So what we want to do is we want to click the FASTA button, and that'll just give us the amino acid sequence as just a string of letters, which is what we want. And so I'm going to go ahead and copy that. And then, so now I think we're just going to do this one first because it's nice and easy. But okay, now we've kind of hit a little bit of a wall. So, we've got our protein. Now we need something to put it into. So I'm going to just search. This site's called AddGene, if you can't tell from the giant sign that says AddGene. This is a repository of plasmids, and it's kind of where I get a lot of my stuff. As in the same way that if you're coding, if you don't need to code the entire thing, if you can just like grab a library, you do. Same idea. Like literally exactly the same thing with biology. If you don't need to make the entire plasmid from scratch, don't. So that being said, whenever I'm designing something, I always check AddGene to see if it's got something that is, close enough, so that I can just, order it, cut it open, stick my piece of DNA in, hunky dory, we're done. So, because we're doing ovalbumin, right? So we kind of have to think this through. If we were to just make a plasmid that just has a promoter and then the protein, basically what's going to happen is once we put it into the yeast are going to start producing that protein. But then you gotta kind of think through, okay, what happens next? Where does that protein go? If we just used a very basic promoter, and protein, it's just gonna make it, and the yeast is literally gonna like fill up with the protein and then die. And the only way to get the protein out is to then pop all the yeast open. Not a fan, personally. This is not a very efficient system. Some things you just can't get away from it. It's the only way to do it. I've got a friend in town who runs a company that has genetically modified yeast to make THC, for example. And they're doing some really, really cool work. The way their thing works is awesome. But in that case, you can't export the proteins that does the biochemistry outside of the yeast because it just doesn't work. So with them, they have to keep it inside the yeast, and then to get the product out, they have to pop the yeast open. A bit annoying, but it is the way it is. Because we're trying to make something brewable, we kind of have to, we really ideally we want the protein to come out of the yeast, so that way you can just kind of harvest the liquid. Like you don't need to harvest all the yeast and pop them open, and you'll get a way lower yield. If you have the yeast export the proteins though, then it'll kind of build up in the actual brewing liquid rather than inside the yeast. And so you'll get a much higher yield because the yeast aren't kind of filling up and dying. And also, you can just, it's easier to harvest the liquid you'll get, and it'll be more closely, it'll be closer to egg white. Like in theory, if you did this right, you would essentially end up with a tank of egg white, which I think is kind of cool. So anyways, this being said, we want egg white proteins out of yeast. And we're going to actually want this same thing for the milk now that I think about it. So we're going to go with a yeast plasmid that will export. Now, there's a couple different ways to do this. The way that I've done this, and actually I can open this up. I've been getting like a bazillion questions about, where the hell did I put it? About the spider silk thing. So the spider silk project is pretty far along at this point. Okay, which one do I want to open? What are you? As ordered. Fuck it, let's go with this one. Yeah, so the spider silk project is pretty far along. And so we actually did the same thing for the spider silk project because I want the silk to export. So we used this plasmid here. I think I managed to grab the empty one because I am a turnip. How the fuck did I manage that? Oh, yep, I opened the wrong one because I'm a turnip. Okay, well, Genome Compiler is great, but it is a little bit laggy, which is not super, super awesome. But it is the way that it is, and so we just kind of roll with it. Yeah, there we go. All right, let's close this. Okay, so this is the spider silk plasmid that we had made, and I have sitting in my fridge. And there's a couple features that I want to point out. I mean, oh, Jesus. So this is this green bit is the silk, and so I'm gonna set this to linear for a second here. I'm gonna go expand view just so that we can actually have a closer look at this. Okay, so yeah, this green bit. Oh, let's not kick my mic. Okay, so the green bit is the silk, and we set this to export because we don't, for the same reason that we wouldn't want the ovalbumin to build up in the yeast, we don't want the silk to build up in the yeast. So what we did was we made sure to choose a plasmid that has a secretion tag. And I believe, yeah, here it is. So this is the secretion tag right here. It's called alpha mating factor. Basically, this is the front part of a protein that would normally be excreted. And so the people who developed this basically realized that if you take the first few amino acids from that protein and stick it on the front of whatever protein you want to excrete, the yeast recognize it as this thing needs to leave, like this needs to get excreted. So if we look at the sequence here, and wait for the lag. Here we go. So yeah, this is the secretion tag right here, and it's pretty. I mean, it's not that big. It's it's less than 100 amino acids. Like it's not very large. The only reason I don't want to use this plasmid again is because of the issues that we've been currently having with it. So yeah, so one of the issues that we're having with this is this plasmid was designed for yeast, yes, but for a species of yeast which I don't like. There are many different species of yeast. The one that most people are familiar with is Saccharomyces cerevisiae. That's the one that is responsible for baking and brewing for the most part. The one that we use for genetic engineering a lot of the time, unfortunately, is a different species called Pichia pastoris. And so this plasmid was unfortunately built for Pichia rather than Saccharomyces. I like Saccharomyces. Saccharomyces is very easy to work with. They grow quickly. They're fairly easy to modify. They're kind of awesome. Pichia, on the other hand, are slow. They're slow. They're hard to get DNA into. It's really just awful to work with. I don't like them. I mean, it's one of those things that, like, I'm very picky about the species that I work with, not necessarily because of certain characteristics, but because of, like, I want the thing that's easy to work with. Like, I even have preferences of E. coli strains that I like working with because some are easier to modify than others. For example, Turbo, very easy to modify. They grow very fast. BL21, utter pain in the ass to modify. Don't like them. So yeah, this is why I'm hesitant to use this plasmid. Also, Pichia is technically edible, but I wouldn't want to eat it. It's not food. So I what I would like is a plasmid that has this same alpha mating factor tag in it that doesn't work in Saccharomyces because fuck Pichia. The other problem with this plasmid is this green bit here, which is the promoter. So the promoter is the bit where the various machinery that will start the process of reading the DNA and then turning it into RNA so that it can then be turned into protein starts. Excuse me. So this is where those proteins literally like grab onto, and then they kind of like scan through until they hit these three letters here, which is ATG. This is the start codon. This basically tells the machinery that like this is protein. Proteins start here. The promoter is just kind of what the various proteins grab onto to get the thing going. The start codon is what the ribosome, so the thing that turns the RNA that comes out of this into protein, starts with. So yeah, this promoter, also not a fan. It's the AOX1 promoter. So it's a promoter out of Pichia. Unfortunately, it's what's called an inducible promoter, which means it can be turned on and only turned on. Like it's not on by default. It only turns on under certain conditions. In this case, when methanol is present in the solution, which is not ideal. I mean, for my work, I just add a little bit of methanol to the media, but ideally, I just want something that runs. I don't want to mess with inducible. I just want something that just go do thing. So yeah, don't like this promoter. this promoter is really the limiting factor of this plasmid. Like if this wasn't here, like if this was a Saccharomyces promoter, this would be fine, but it isn't. So real annoying. Be that as it may. So what we're going to do, we're going to go over to AddGene, and I realize that I am rambling a lot and we're not making a lot of progress, but I am also trying to like cover a lot of information. So we're going to go alpha mating factor, and we're going to just look up, see what we got. So we go to plasmids, and we're going to see what we have available. So we want an empty backbone. So plasmid type. There's like a short form for alpha mating factor. It's the AF1. Let's look this up. Alpha mating, factor. Hit caps lock. So a lot of this is just kind of like looking up, AMF. That's what they call it. AMF. So yeah, a lot of bio stuff is just like knowing what to look up, and especially with AddGene, like sometimes people just label their stuff in just stupid ways. So a lot of the time is, see, this is what I mean. Alpha mating factor right here. Type 3A secretion signal, and it looks empty to me. Yeah, there we go. Alpha mating factor secretion signal, lambda terminator. But is there a promoter? All right, so now we do a little bit of research. I'm going to open that one up into the side. No EA, I don't know what that means. Kex recognition site. Inducible, we don't want inducible. So it's basically just like shopping around, trying to find the thing that you want. Species, Saccharomyces, let me guess, it's the two that are at the top of the list. It is actually, it's the inducible ones, which is worse. Okay, well, let's open up this one that I opened up in a new tab. Does it say what the promoter is? That's really the thing that... Okay, so this is actually, I think this is important information. So I'm gonna stop and just talk about this for a second. So this is some of the information that it gives you about the plasmid, because I mean, like just looking at this, like it's pretty sparse. Oh, that's exciting. I actually just noticed something really awesome, and I fuck with this. This is great. Okay, so let me just go over all of this just so everybody's kind of on the same page. Okay, let's start with, this is the plasmid page. This is all the information about the plasmid. So it tells you what's in here. The main thing being the alpha mating factor secretion tag. This is what we want. This is very good. There's other features here. This bit right here, the CMR, is an antibiotic resistance, and it won't work in yeast, but it will work in it'll work in bacteria. So one of the things with plasmids is they're almost always built to work in two species, E. coli and then the species that you actually care about. And the reason for this is E. coli are very easy to modify. So if you're having issues getting DNA into something, the first thing that you'll always do when you get a plasmid, I mean, and also like AddGene sends you the DNA not as pure DNA in water. They send it to you in bacteria. Like that's just how they send it. So that way you'll never run out. Like the first thing that you'll do when you get this plasmid in the mail or whatever is you'll streak it out because it's in bacteria. And then you can just grow that bacteria to whatever density you want and, extract DNA out of it, this DNA. And then you have all the DNA you could ever need or want. So in this case, the two pieces of information that it gives you, so it says growth in bacteria, it's this thing. What strain does it like working in? This doesn't really matter. It'll work in anything. You can ignore that. The important thing, though, is these two pieces of information. Chloramphenicol, which is the type of antibiotic used to select for, and high copy. High copy means basically what it sounds like. There will literally be lots and lots and lots of copies of this piece of DNA per bacterial cell, so that when you go to extract DNA out of those bacteria, you have as much DNA as physically possible. These are all good things. Chloramphenicol is fine. It's a really nice. I say it's a nice antibiotic. It's a nice antibiotic to work with because it's quite potent. So you very rarely run into situations where you've got shit growing on your plate that you don't want. So as these things go, this is good. The reason I got excited when I opened up this is called Plasmid map, by the way. We've now seen several of them, but that's what this is. You'll notice this one's pretty sparse. It's also very small. So it's a little under 2,000 base pairs, so 2,000 DNA letters. That's very small, like very, very, very small. Most of the plasmids that I'm used to using are massive comparatively. So yeah, this is tiny. This is absolutely tiny, but it's tiny because it's basically empty. Like there's very little in here. So the reason I got excited was because of this right here. It says NDEI. These little strings of characters are what are called restriction sites, and they denote which enzyme. So a restriction enzyme is a special type of enzyme that homes to a very specific sequence of DNA and will cut at that location. So NDEI, the reason I'm excited about it is because I just bought some. So I have a brand fresh tube of it sitting in my freezer. So if we look this up, NDEI, restriction site, and then we go to, let's go to Thermo, because that's where I bought mine. So this is the series of letters, CATATG, and the little arrows here, it might be a little hard to see, but it cuts here and here. So this is what's called a sticky restriction site, which basically means that when it cuts the DNA, it leaves this like little dangly bit sticking out. And this actually leaves a spot for, like it leaves it's easier to stick things back on here. So if you cut something else that has an NDEI site and you literally like mix them in a tube, one end, like the piece A, this piece will stick to this bit. I never use restriction sites this way. I use a method called Gibson assembly, which we're going to talk about in a second, because I think I think we're going to go with this plasmid because I think it will, I think it'll be helpful. I think it's a good learning experience, and also it's mostly empty, so we can kind of add whatever we want, which I like. So yeah, we're gonna go with this. Let's do it. Let's do the thing. So I'm gonna go ahead and view all sequence. I really should have. That was stupid. I was in the window that would already give it to me. Okay, so there's also different flavors of file that you can download. I always go for the SnapGene one because SnapGene is the other. It's an alternative to Genome Compiler. It's the one that actually a lot of people are used to, but it kind of formats everything really nicely. So sometimes if you don't download the SnapGene file, like you'll have a chunk, like the letters will be there, but like, for example, like the CMR bit might not be labeled. So it's just this like empty void, which is awful. So anyways, let's get going. Okay. So I'm going to close the spider beer because I don't need it. I don't need, yeah, I don't think I need this either. Okay, let's bring this in. Okay, import file. Let's grab the new one. And milk and eggs. I've already made a folder for this because I did a little bit of prep in advance. And I see a couple of people are getting bored. Well, strap in, because this is what bio is, man. It's like coding, except slow. And, you do a lot of planning, and then you wait 6 months while somebody makes your DNA for you. Okie dokie. Now, this is what I mean. Genome Compiler is inherently laggy, so sometimes it just takes a minute. You just kind of got to be chill. All right, it's open. I'm going to close this other one that I had open. It's got a lot of interesting things in it, but I can live without it. Actually, you know what? Maybe I'm gonna leave it open. There's some features in there that I kind of want. So now we kind of have two options, right? Option the first is, do we just want to take the alpha mating factor out of this plasmid and then put it into a different one? Or do we want to add a whole bunch of stuff to this plasmid? And I'm not entirely sure which way I want to go with this one. And I'm kind of thinking, oh, you know what? Yeah, let's go with this way. Okay, so let's go back to actually this one that I had open before. I'm unfamiliar with this plasmid. This is the first time I've used it, so we're gonna just kind of go for it. It's T7 promoter. That's interesting. Okay. Wow, they've really crammed a lot of crap into this. Multiple cloning site, but I don't know if this is the plasmid I want to go with. Let's just have another little look-see on AddGene. Wait, actually, I had something open. Where did I put it? Yeast vectors. Here we go. Integration plasmid, centromere plasmid, obligating plasmid, some plasmid. So just have a list. So the thing I'm looking for right now is I want to find the best plasmid for this. Because this is one of those, like, it's better to take your time and design this well now than it is to try and fix it later. Because with bio, you do a lot of like prep in advance, and then you kind of go from there. You don't want to like, once you've bought the DNA, you're like down $1,000. So fixing it is just a nightmare. So if you can avoid that, that's ideal. So I'm looking at this thing, and yeast vectors, come on, show me a fucking list. No, really? Nothing? God damn it. This is frustrating. I'm just trying to find a good basic yeast plasmid that we can work. ADH1. ADH1, I don't really like ADH1. ADH1 is alcohol dehydrogenase. It's which is referring to the promoter, I believe. Let's have a look at this. Okay. I assume that's this. Barf. Leucine. Okay, so there's kind of a list of things that I'm looking for, and this is sort of the reason why I'm like waffling and flip-flopping here, is I want basically 3 things or 4 things. So obviously it needs to, like, the plasmid needs to work in yeast, specifically Saccharomyces, because I'm not messing with Pischia again if I can avoid it. Thing number 2, it needs to have some way to select which yeast have actually taken in the new DNA when you put the DNA into them. And what this plasmid that I happen to have open right now uses for this is this orange bit here. It's basically, there's certain amino acids that yeast just can't make. And so if you grow them on media that is missing that amino acid, literally the only ones that'll grow are ones which have taken in this DNA, because it contains a little piece of DNA that lets it make that amino acid, basically. So I hate this system. The reason being that it's like $300 or $400 to buy the media that does this, because you essentially have to add individually every single amino acid except the one that you're using as the selection agent. And then you need the yeast nitrogen base, and just it's awful. It's fucking awful. I hate doing it. I refuse. So I always use plasmids that use an antibiotic as a selection agent because, well, it's cheap. It's super super cheap, and it's easy, and it's effective, and it's just nice. So why would I not? And then there's an argument to be made about, maybe this could be like, maybe are you releasing antibiotics into the environment, or resistance into the environment? But the reality is, like, literally every person is covered in bacteria that are resistant to every antibiotic we know. Like, there's no such thing as a person who's free of this. They've found like vancomycin resistance in isolated tribes in the middle of Africa. It's not going to happen. So I'm not really super bothered by this. And there's an argument to be made, but I just don't care. So okay, how do you? I'm going to try looking for the alpha mating factor by just grabbing the... Oh, come on. Just give me the fucking... I need the Greek letter so I can look this up properly, because it does actually search for this. I need the just normal alpha. Where is this? I'm sorry this is taking so long. Let's see if that shows anything. Surprisingly, yes. No. Alpha mating factor? Okay, I had a list of promoters. Maybe we'll just search by promoter. Actually, that was what I'd done before. Repressed by ethanol. We don't really... See, the reason I don't want to use an ADH1 promoter is that it is this thing. It says it's repressed by ethanol. So basically, as the yeast start growing and producing ethanol, because they're yeast, that's what they do, they're inevitably going to stop producing the protein you want, which is not helpful. So I'm just trying to see if I've got another one that could work. So TF1 is another option. And again, I'm just going to keep sort of waffling on this. Once I find the plasmid that I'm like comfortable using, this whole process is going to go a lot faster. I'm just I'm waffling until I find something good, because I want to make sure that I don't have to do any more work than I absolutely have to. And yeah. Oh shit. There we go. So yeah, there's two things I want. I want TF1, which it should be a fine. Yeah, here we go. So this would be perfect if it didn't have a bunch of other crap in it. Cas9, dual reporter backbone. Ooh, M cherry. That's exciting. Let's just have a quick little look-see here. What do we got? Okay, can resistance. That's exciting. M cherry, TF1 promoter. Okay, I'm liking this so far. TRP1, I don't know what that is. I assume it's an amino acid. There's a multiple cloning site. There's a multiple cloning site for what? Okay, I'm going to open that one, come back to it. I got a thing. What was that? Yes, I can make a video about genetically modifying plants. I've been working on it for a while. It's just hard. Like modifying bacteria, very easy. Modifying plants, more of a pain in the ass. So really, what I want, vector type, shit. What's really frustrating is like this would be perfect if it didn't have other crap in it. Let's just yeast can. So I'm gonna, rather than going for the promoter, I'm gonna just go for the restrict or antibiotic resistance. You'll notice that I keep looking up can. This can MX thing means it gives the yeast resistance to G418. Yeah, which I have lots of, and G418 is very cheap. So if I can use that, that makes my life a lot easier. Ooh, integrating. Hello, what do we got here? Empty backbone. I think we might have finally found a winner. I mean, I'm going to have to do a little look-see and see what we've got, but how does this work? Why is the multiple cloning site over there? That's a very weird... Damn it. Okay, so there's different flavors of yeast plasmid that kind of do different things. The ones that I like, yeah, this is the same thing, are what are called integrating plasmids. So basically, once you put the DNA into the yeast, it will actually integrate itself into the yeast genome. So it makes it much more stable, and it'll last a lot longer. Is this the same thing but with an insert? No, see, like this is the thing that's confusing me now. You've got a multiple cloning site, which is where you actually stick the protein you want, like right here. But the promoter is in a weird spot, and I don't know what to do about that. Vector types. Expression. Definitely yeast. Empty backbone. That's what I want. Okay. I'm just trying to, I'm getting closer. I'm getting much, much closer. I'm just trying to zero in on getting exactly the right thing, because there's nothing worse than going through a whole, like, designing session just to find out you grabbed the wrong thing and you got to start again. And that's no fun. Target sequence, HO locus, can selection. Where's the multiple cloning site? Middle of fucking nowhere. That's not helpful. Tat can. Ooh. SP6 promoter. Okay. Okay. Chemomycin resistance. That looks like a multiple cloning site. Let's look up this promoter really quick. SP6 promoter. Is this constitutive? RNA polymerase. That sounds aggressive, which I'm kind of into. Oh, no. Boo. That's annoying. Well, I'm gonna open this up and have a look at it just to see. Empty. I do want to make sure that it's... That's weird. Okay, that's very strange. All right, let's open this up, have a look. I mean, like, this looks like it would do the thing that I want. I just don't know. I don't know. Also, I mean, the RFP here is not ideal because it would make red egg yolk um, and or red milk, which I mean sounds entertaining, but I don't know if people would be into that. I'm into it, but I don't know, not ideal. CCRA. Like this would be perfect, except that the multiple cloning site is in totally the wrong location. I mean, we might be able to work around this. MSCI. What is this? Is it oh god. See, this is what I don't like seeing. If you go to buy, oh, actually, it's not so bad. It's 200 units. It's 5 unit per microliter. That's actually not so bad. So yeah, when you go to buy a restriction site or a restriction enzyme, some of them are really expensive. Like the weirder and rarer it is, and the less used it is, the more expensive it's gonna be. So I kind of got wigged out for a second there because I saw 5 units and I was like, oh god, they're gonna charge $400 for 5 units. But actually, it's 200 units for about $50, which is not actually the end of the world. That's actually fairly cheap, all things considered. It's not what it's maybe not the thing that I would like to go for. Given better circumstances, but it's one of those, if I were to get this plasmid, which I think, where the hell did it go? It's this one. Yeah, here we go. Yeah, if I were to, say if I were to get this one, I could make some changes that would make that a little easier. Like I could stick a multiple cloning site right here, and it would only cost the like $50 to buy that one enzyme. But yeah, I mean, if we make red milk and eggs, in a future stream, maybe we can add like strawberry flavor, so it's like natively strawberry flavored milk and eggs, which sounds ridiculous, but this is the nice part about genetic engineering. You can do whatever you want. I mean, the other option is you could try and cut the RFP out, which I don't actually think is a very good idea. But yeah, again, the multiple cloning site is in totally the wrong area. Not really sure about this, but it says dual reporter plasmid. So presumably, this plasmid was designed so that you could stick something in this multiple cloning site, and oh, it's got a YFP in it too. Oh god, that would look so weird. It'd be like orange. And how red are we talking? Like very. I actually I have some in my other fridge. It's really red. Like really, really red. It's pretty obnoxious. It would not be appetizing. So unfortunately, I think we might be back to the fucking drawing board because I really want something that isn't gonna need 82 and AEI. This could work. I don't see a terminator though. I mean, this looks like a multiple cloning site. This is so stupid. Why is finding a fucking yeast backbone so hard? I've spent like over an hour on this. I mean, like, I'm just gonna open this up to the comment section. Like, I've got my eye on the comments. If anybody can find me a yeast backbone, because I know we've got some bio people hanging around. So yeah, if anybody can just find me a yeast backbone, I want kanamycin resistance. I want a constitutive promoter for Saccharomyces. If anybody can find that and link to it, I'm just gonna keep an eye on the thing. I'm gonna keep looking, but like, my patience for this shit is rapidly dwindling. Because again, I really don't want to use Piscia. I hate Piscia. It's a stupid organism. Like this looks like it would be perfect. Why can't it just? Right, like they're putting the promoter in the worst possible spots. This is very frustrating. Yeah, I'm not seeing anything in the comments, unfortunately. Right, like I've passed this one like 3 times. Maybe they've got something. Yeast can backbone plasmid. Maybe they just list one. That'd be good. Various markers, promoters. Okay, I'm just gonna keep browsing a little bit. We're gonna just see what we find. Contents of kit. No, this is not what I want. See, now we're talking. Hopefully one of these is gonna have something useful in it. Uracil. Garbage. I can see that fucking uracil from here. I really don't want to have to add the kanamycin resistance myself. It's just so annoying to do. Fuck. Okay, well, what did I have open already? What is this? Where's the promoter? This is the thing that's confusing me. Like, there's a multiple cloning site here, but there's no useful promoter. Nothing in the comment. Oh, hello. What's that called? Thank you, Todd Gore. Okay, let's look this up. Hopefully it doesn't dip in the chat before I can type it in. PFA6A-CAN-X6. Can resistance. SP6 promoter. T7 promoter, but where would you... I mean, that looks like a multiple cloning site. PCR-based gene targeting. I mean, it's close, but it's not quite what I want. FA6A. God damn it. Yeah, I see that, Todd. Thank you. I'm looking at it now. I don't... It looks like it would be perfect, except that it's got this SP6 promoter as the only thing. Like the TEF promoter is in front of the resistance, which is obnoxious. Does SP6 promoter yeast? I'm gonna look this up, see if it works. In vitro transcription, general expression. Used for in vitro transcription drives either the... Yeah, SP6 requires a weird RNA polymerase to work. So this it's really close. I really appreciate this, but it's not quite. It's not quite what I want. This is the issue. This is the thing that I continue to like bash my head against. And I'm not really sure. And also, yeah, it's a bacterial promoter. It's not in yeast. Mammalian. Anybody else got some suggestions? Thank you, Todd. Unfortunately, it's not quite right. Yeah, man, this is really fucking me up a whole lot more than I thought it would. I thought this was gonna be like, this should be the easy part. But here we are, an hour and 15 minutes into the stream, and I still don't have a fucking backbone. This is just beyond frustrating. Sorry this is taking so long, guys, but we're gonna get this sorted, and then I'm gonna be able to stop bashing my head against this one stupid thing. I've looked this up like 4 times. Yeah, so anyone else who's got, just take the red milk plasmid. I like it. I like the, I like the idea of the red milk plasmid, I do, but, I just think it's a bad idea. What's this one? I'm just gonna keep checking until I find one. Cerevisiae. Okay. Hello. What are you? Gap promoter. Why the fuck does it say that it's a different promoter than what's in the map? Like this is the things that just drives me insane. AddGene 1786. 1786, what's that one? Oh. For fuck's sake. God damn it, Paul. I mean, I could remove the red from the red milk plasmid. Unfortunately, the red milk plasmid also has yellow. Like it's a double reporter plasmid, so it's not just red, it's also yellow. Which is so much worse. Where did it even go? What did I do with it? Oh, it's here. Yeah, see? So this is the red milk plasmid that everybody in the comments is talking about right now. Thank you for throwing in money to tell me that the red milk plasmid is a good idea. I appreciate that. But look, so here's the problem. I'm not opposed to the red milk plasmid per se, except there's 2 in here. It's not just red. It's also yellow. So there's a red and a yellow. So it's completely unsuited to doing this. It would be fine if it was, yeah. So like even if I cut the red milk out, it would still do yellow and it would just look awful. I mean, it'd be fine if you're making cheese, but like fluorescent yellow cheese, yeah, not AddGene, PSmart, HCCam. Oh, okay. Wait, shit, where did it go? Oh, see, it's more red. I don't want anything that glows, god damn it. I'm appreciating the suggestions, but can we please focus on one that doesn't frickin' glow? 46815. I'm gonna just check all these. Like all the ones that you're suggesting. Hello. This looks perfect so far. Where's the... Oh my god. Who sent this one? Where did this just come from? Sassy's Tripper Sniper. Oh my god. I think we found it, boys. I think we got there. Okay. Holy shit. I think this is actually serviceable. multiple cloning site. It's got the correct promoter. It's got the kanamycin resistance. It's got, oh my god, this is amazing. Dude, Sassy, you are the bomb. This is it. This is exactly the thing that I needed. Holy crap, I am so sorry that it took this long to get here, but we got there, boys. Oh my god, I hate biology sometimes. It's just the worst. Okay, cool. Well, I'm gonna close this crap because I don't need it. Wow, that was so obnoxious. Is it gonna close? Or is it just gonna lag some more? It's gonna lag some more. That's exciting. Okay, import file. Wow, that took, frankly, way too long. Just way too long. Okay. Well, thank you, Sassy. You're the best. That just made my life so much easier. I can work with this now. 46815, I believe, was the one I just. Where did it go? Okay, I'm gonna close a lot of this other crap now that we've actually found the thing that we wanted. Finally. No, I don't want this one. Oh no, wait, shit, that was the one. Damn it, that was the one I wanted. Okay, you know what? Who cares? I'll just 46815. Yeah. Yes, if I make some, I will send some to Sassy. You've earned it. Oh my god, that was so frustrating. Holy crap. Man, this is so much harder to see. This is kind of nice because, like, I think this has been a really great demonstration of just how frustrating this can be. Like, just zeroing in on the right thing can be very, very hard. It's fine. I was only checking so that I could open the right thing. Okie dokie. I just want to have a look at this in circular view and make sure that everything imported properly. And by God, it did. Oh my. But that was so frustrating. That was just so goddamn frustrating. Have I mentioned how much I dislike biology? Okay, finally, we can finally get some frickin' work done. Okay, now that we know that this is the plasmid that we want, we can actually get designing. So let's just give her a little clickaroo over here. Cool. This already put. That's a weird thing to have in there. Epitope tag. I don't know why that's in there. That's very strange. See, this is what's really interesting now that I'm actually zoomed in on this is it's got the T7 promoter as well. So anything that we stick into this spot will not only be produced in yeast, but it'll actually also be produced in whatchamacallit, in bacteria. So that's actually really cool. Man, this was a great find. Sassy, you the bomb. I'm really quite pleased with this. You've found literally all of the things that I could have ever wanted in a plasmid. Oh, man, that was really stressing me out. Okay, well, we're an hour and a half in and we can finally get started. Shit. Well, it's nothing if not realistic. Just to give you a back, this particular promoter isn't important so much as I need a promoter that is yeast specific that'll work in a variety of kinds of yeast. I also needed it to have kanamycin resistance because the same gene that codes for kanamycin resistance in bacteria also codes for G418 resistance in yeast, because yeast are naturally resistant to kanamycin, so you got to use G418. It's slightly different. Yeah, E. coli milk, delicious. But anyway, so now we can get going. So I'm just trying to make sure that there's no fucking start. Yeah, there it is. See, okay, here's a problem. So, well, it's not really a problem per se, but okay, it's not. Come on, highlight the G, man. There we go. So there's an ATG here. So this is where this is actually going to start, which is a little annoying. But I can actually deal with that. That's fairly serviceable. Really, I want to highlight this SAII or SAL1, depending on how you want to pronounce it. Okay, so now I'm just debating, do I want to do SMI or SAL1 as my restriction site? And I think I'm going to go SMI. I wish I'd had the presence of mind to have this open. But I actually have a list of restriction enzymes that I already own. So I'm just gonna open that up really quick. And I'm opening it up in not the screen that I'm sharing with you guys, because this is not a document that I can share, because it's got some ish to it. And also it's in Google Drive, and I don't feel like broadcasting this this email, frankly. Okay, so I've got my list open, so I'm just having a quick little look-see to see which of these two restriction enzymes I have. And technically it doesn't really matter, because this is not... Wait, what am I doing? This wouldn't even matter. I would be sending this to the synthesis company, and they'll just cut wherever I tell them. But I just want to have a little look just to see what I've got, because it's... Okay, yeah, I've got SAL1, and do I have SMI? I do not have SMI. SAL1 it is. Okie dokie. So SAL1 is here. So let's... I just want to see where exactly it cuts, because you don't want to mess this up. Okay, so it cuts at the G. That's important. So it cuts there. Is this only showing me one strand? It is. Okay, that's obnoxious. So let's go ahead and turn on the minus strand. Because remember, DNA is double-stranded, so yeah, that was a little... Okay, that's so much better. Ugh, so much better. Okie dokie. So, the first thing I'm going to do is... I'd really like to have another folder, maybe, but I'm going to go ahead and duplicate... I'm going to duplicate this. Just save as. Um, 46815, 46815. We're gonna go plus or with Ovalbumin. Okay. So I don't like to, and this is something I learned from my fantastic lab partner Vesta, who may or may not be listening, not sure. But yeah, you never mess with the original file. You always leave the original file alone, so that you can come back to it if you fuck something up. So now we're in a duplicate of the file. Like we've got the original empty plasmid, and so now we can actually kind of get to work. So now I can finally bring up the ovalbumin. We're gonna grab this guy, copy. Now we're gonna go back to Genome Compiler. Okay, so when I go to make DNA, like when I put things together, I use a process called Gibson assembly, which is a little different than what some biologists use. I mean, it's not that weird. I like it because it is very powerful, but it also has its quirks. So one of the quirks is if you're gonna Gibson something, what ends up happening is it chews back the, I want to say I can never remember this. It's either the 5' or the 3'. It's this end. So what you would do, right? If you were assembling this, right? Let's say that you've already got your insert. So in this case, it's going to be ovalbumin with the alpha mating factor stuck together, right? So let's say you wanted to put that piece of DNA in here. If you were doing this in a more traditional, I believe it's called Golden Gate assembly, you would cut here with the cell 1, and that would open up the DNA. So you've got a dangly bit here and a dangly bit here, and they're kind of like, well, it'd be like this. So your dangly bits get cut, and they're kind of just floating around in the breeze. Now, if you were doing this with Golden Gate assembly, you would just make sure that your insert also had the matching dangly bits on it. So you just would cut it with the cell 1, and so the dangly bits would line up and stick and match, and then you could just close it up. Gibson does not do this. Gibson chews back the 5' end. So literally, like from this T here, it'll just chew back like 20 to 30 amino acids, nucleotides, all the way to like back here. So instead of just like 4 letters that are dangly, now you have this entire sequence that's exposed and can be used as like a sticky targeting section, which is fantastic because it means that things stick exactly where you want them to. However, it does come with the caveat that it's going to cut off this little dangly bit because it chews it back. And because there's nothing to match on the other side because of the way this cuts, like if we were to cut with the SMI, it's blunt, and so you don't lose any letters. But with this, if you're cutting with cell 1, because it's a sticky cut, you do lose some letters, which is a little annoying, but it is the way that it is. Okay, so that being the case, I'm going to go ahead and we're going to just assume that they use Gibson assembly. I don't actually know if they do, but the nice thing with the company that I use, which I will not mention, because there's lots of different companies, and the company I use is fantastic, and I don't like sending huge amounts of internet traffic to them and freaking them out, because I really don't want to lose my account because they're really good at it. But there's all sorts of different gene synthesis companies. A lot of the time you can send them the empty backbone that you want to modify, and then you tell them which bit of DNA you want them to synthesize, and then be like, put this piece that you've synthesized into this spot in the backbone, which is basically what we're going to do. So without further ado, so considering that this is going to be, we're going to assume that they use Gibson assembly like I do. So we're going to assume that this bit is going to get chewed off, basically, just because of the way that the enzymes work. So I've already gone ahead and copied my ovalbumin sequence, and this is just in amino acids. So, so we just copy the amino acids, and we're just going to go ahead and paste. Cool. So it recognized that I'm pasting in amino acids, which means it needs to translate them. So we're going to hit a translation table because, we're going to go with codon usage, and Saccharomyces cerevisiae. So I'm going to just pull this up really quick just so I can show you guys DNA codon chart. Okay, so this is the DNA codon chart. So just as a quick refresher for those of you who are not biologists and who are just trying to follow along, I'm going to grab one that actually uses T's. So the DNA is broken up into 3-letter sections called codons. And this is these are all the like theoretical codons. These are the so yeah, theoretically these are all the codons, but every organism does not actually use every single one of these. So you'll see that like for proline, for example, there's 4 different codons that code for this amino acid. Most organisms will prefer one or two of these over the other ones. And there's a mechanism inside the organism that will like physically prefer these like a specific thing. So when we're using a codon table, you'll see that I've set it to yeast Saccharomyces cerevisiae. It's going to use the codons that Saccharomyces specifically happens to like using. If we just used whatever random codons, it would still probably work, but our yield would be crap. So we're going to set it to Saccharomyces because this would be expressed in Saccharomyces. So this way we should get maximum ideal yield, which is great. So we're just going to click OK, and then it should just paste that in. And it should, unfortunately, I know the comment section is upset about this, but yes, it's going to get rid of the dangly bits. Wait. What the hell? The dangly bit survived. Just by a quirk of the protein, it put the dangly bits back. I know this is exciting. I know everyone's gonna be very happy about this. Okay, well, anyways, so you'll see that it's now pasted in the protein. The dangly bit is still gonna go away probably, but I'll put it back in afterwards. So I'm gonna just move over to annotations. It's already highlighted. So now I need to tell the program. You know what? Screw it. I'll do it this way. Annotate selection. So now I need to tell the program, because I just pasted in essentially a random assortment of letters, so the program doesn't know what to do with this. So I need to tell it what it is. So I'm going to go and I'm going to set it to CDS, which is coding sequence. I'm going to set this to circular so you can see the change happen. And so you'll see that now it's rather than just like a random bit, it actually knows what this is. So I'm going to give this a name, Ovalbumin. So now I know what it is. And I'm going to give it maybe a different color just because there's a bunch of other orange things already. So, the dangly bits restored. Yes, I will make sure to put the dangly bits back. I'm going to make this pink because why not? It'll stand out. It'll be easy to see. So yeah. Okay, now we can go back to here. You'll see that for those who were wondering, yes, the dangly bits are still in fact there. Just as a quirk of the sequence of this protein, it happened to contain all the right letters. So when I pasted it in, it was still there. So that's fun. I mean, for most of the people who are like, you should have studied this in university. University did not teach me this. This was learned through doing literally what I'm doing right now, and through making stuff like this. University taught me nothing. Like I did 3 years of it before I was like, yeah, no, I'm done. Can't. You're teaching me boring ass population genetics, not genetic engineering. I want to do genetic engineering. So anyway, so yeah, dangly bits restored conveniently. So that actually means that, in theory, they wouldn't have to use Gibson assembly. They could just, put it in normally. So that being as it is, we now have, theoretically, by some fucking miracle, we actually have this is almost done, basically. So this is what I meant when, once I have the plasmid, this will all go a lot faster. We now have a plasmid that will express ovalbumin. So if we were to send this off to a synthesis company and be like, make me this, please, we would have yeast that make ovalbumin. But now we return to the problem that I mentioned way back before, which is if we just had them make this, the protein would build up inside of the yeast, which is not ideal. So, what we got to do is we need to add that excretion tag. And the nice thing is, because this would be synthesized, that is to say, they'll just print whatever DNA I tell them to. We're not limited by the older school methods where, we've got to cut this out and PCR the thing that we want from something else, and then do an assembly and blah, 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 blah. They can just synthesize this entire insert all as one piece, and then just shove it into the plasmid, call it a day. So that being the case, this other plasmid that we had opened before, which contains the alpha mating factor that I'd been kind of waffling about using, I can just like highlight the whole thing and go copy, and then come over to here, and I'm actually going to make another non-destructive edits. So I'm going to save this as with ovalbumin and secretion tag, because there's nothing worse than than doing making a change, realizing it was a mistake, and then having to go back and fix it. Okay, cool. So now I have two open. We're now in this one with but now we got to add the secretion tag. The reason I did that is just so that I can boop back to the other thing and make sure that the dangly bits are maintained. You wouldn't download a Plasmid. Yes, I would. I always love those commercials. They're like, you wouldn't download a car. What is that an option? Can we do that? Is that like yeah, maybe next year I'll do the April Fools, the genetically modified cat girls. Anyways, okay, so we've got the alpha mating thing copied already, so I'm just gonna go ahead and paste it in standard code. Keep original sequence. Pasty paste. Cool. Okay, so it has pasted that in, and once again, by a weird quirk of, is that really right? Have I managed to keep the Sal1 thing twice with no effort? Because that's just insane. I mean, it looks right. Okay. Cool. So, for those wondering, the dangly bits are still there. Just by some freak of ish. All right. Wow. I was not expecting that at all. Okay, cool. So, we actually basically, this is done. I'm going to give this a little look-see just to make sure that it all looks right. But yeah, no, this is done. So the egg plasmid is looking fine. Okay, so let's just go over this. I'm just checking some off-screen stuff for a moment. Cool. Yeah. Okay. So, let's just go over what we've made now. I'm going to minimize this so we can get a little bit of a bigger view. Okay. So, we started with an empty backbone plasmid which could be purchased from AddGene. Then what we did was so this green bit right here is the TEF1 promoter. This is the thing that I'd been searching for, which Sassy, being the bomb, found for me. So we took this empty promoter or plasmid, and then we basically pasted in two things. This pink bit is ovalbumin, so it would be from it's chicken egg white, basically, is what this is. And then the bright blue bit is a secretion signal so that the ovalbumin does not get trapped inside of the yeast. It instead would get excreted into the solution around the yeast. So what's nice about this is you could basically brew like a gallon of this stuff, and just like pour off some of the brewing liquid directly into your baking, and just bake with it directly. And this is nice because it's Saccharomyces cerevisiae. It's already freaking, like, you could do this in baker's yeast. So like you could make a strain of baker's yeast that produces ovalbumin, which is kind of cool. Like if you just kind of made like a sourdough type thing, like if you so you could like modify the yeast and then make like a sort of pseudo sourdough using this modified yeast, and it would get goopier as it goes, which is kind of fun. And the nice thing is this is pretty small, so I'm gonna just do a little checkaroo here. Let me close this. So this is showing me a length of 1,434 base pairs, and it actually highlighted a little bit too much. So I'm gonna click off and just see if I can highlight just the correct parts. There we go. Okay, so yeah, 1,425. So this is not a very large insert. This would not cost a lot to have synthesized at all. The place I like using they charge about 18 cents per base pair. So 1425 times 0.18. Yeah, so this would cost about $250 to make, give or take, which is nothing. Like when it comes to getting things synthesized, like getting DNA synthesized. So this is actually really cool. I would say this is 100% doable. Like this is 100% synthesizable. And yeah, I think this could be really great. So I may actually end up getting this synthesized. I might do a little fundraiser, but it's within a price range where I might just do it anyway. Just because it's so cheap, like $250 for a thing that, you know, I can brew my own egg whites, like that's pretty fucking cool. And of course, you know, this is, so I'm gonna, once the stream's over, I'm gonna put this plasmid up on GitHub so that anyone who wants it can play with it. I had a question. Why aren't you factoring in the ovalbumin sequence into the cost? I am. The pink is the ovalbumin. The only part you pay for is well, I mean, so you pay to buy the plasmid, and there's a one-time fee to submit the empty plasmid to the synthesis company, so they just have it in stock. But then the only thing you pay for is the actual synthesis of the gene insert that you want in the plasmid. The assembly and however long it takes them to do everything else is basically included. So, I mean, all things considered, it would probably come out to about an extra $100, maybe so, or maybe two. So, like at most, I would say the cost of this, including the submission fee, and I always pay a little bit extra so that they send me just an enormous amount of DNA. So it's usually 10 times 100 micrograms. So it's 10 vials of 100 micrograms of finished plasmid DNA, and a pre-transformed E. coli. So they'll take this and they'll stick it into E. coli. So should by some weird like fluke of fuckery you end up running out of all of that DNA that you paid extra for, you've got it in bacteria so that you can just grow more, which is annoying, but it is the way that it is. And, you know, it doesn't cost very much. So realistically, the price of this project would be about $400. So yeah, that's actually not bad. Like so the Spider Silk project cost me about, like 900, give or take. Which, you know, felt like which felt like a lot, but wasn't so bad, especially considering the amount that I was spending on just doing the lab work of just trying to put it together myself. So, you know, that's not so bad. That's really, really, really not so bad. So yeah, about $400, and we could make egg white. So cool, we're done. By some fucking miracle, we are actually done the first part of this. So, yeah, so ovalbumin is about 55% of egg white. The rest is mostly water. Show that. So yeah. So basically, right now we now have a plasmid that, if you were to make it, would make egg whites. You could brew egg whites, which is pretty cool. Which is, I mean, this is the reason that I wanted to do this stream, because it shows that, like, you know, it definitely took a little bit of time to find the right plasmid, and that always like to start with, and that always does take a little bit of time. But in the span of about 20 minutes, we went from having correct plasmid to having finished plasmid, because it's not hard to put together. Okay. Yeah, right. Like, I mean, I'm seeing some comments like infinite egg whites for $400. Yeah, infinite egg whites for $400 is pretty damn good, especially when all you've got to do is feed it sugar. And like that's a great protein source. Like there's all kinds of things that are great about this plasmid. It's one of the reasons I love genetic engineering. Like people get all wigged out about like, oh, it's genetically modified. Genetic modification is amazing. Like I don't know why people get all fucked up about it. It's like there's nothing dangerous about it at all. And to the question of how do you separate the whites, I mean, you shouldn't need to. Like the whole, like it's gonna taste a little yeasty, but if you really wanted, you could like spin it down in a centrifuge to like get rid of all the yeast cells, and then the protein would just kind of be what's floating above. But if like the plan was to bake with it, like you wouldn't notice. You could just use the fermenting liquid directly for baking. You wouldn't notice the yeasty taste because you're baking. It's already going to taste like yeast. And so yeah, that's the thing. Anyway, so yes, to the question, are we going to do the milk part as well tonight? Hell yes, we are. I am committed to getting this done, and it's not like any of us have anything better to be doing. We all should be in our homes. So I might take a little break in a minute just to make another cup of tea, but like you bet your ass we're making the milk one too. Okay, cool. So I'm gonna go ahead and save this because we've made some changes. And now that that is saved, I'm gonna actually close this. I'm gonna leave the alpha mating factor one open, and I'm gonna just open up the basic plasmid again. Shit, I gotta set it to single project view. And now we can get going on the milk part, which this one's going to be a little more iffy, but it's not going to be much harder. There's definitely going to be some stuff that we're going to have to include. The protein is going to be much larger. It would be a much more expensive synthesis, but all things considered, it wouldn't actually be so bad. Okay, we're now back to okay, cool. So now we're back to empty plasmid where we started, just like in the beginning. So first things first, we're going to do exactly the same thing as we did last time, which is click Save As, milk and eggs, 461, 815, with... let's go with beta casein first. Dear beta casein. I saw a question go by. How does one get started doing this? You just start. There's no, you pick a project and you just start. There's no class. There's no nothing. You buy a copy of the manga guide to molecular biology. You open up Genome Compiler, you pick a project, and you just bumble your way through it. There's no, like, it's just the way you do. Like, there aren't great classes to teach you how to do this. Like all the people I know who are just stellar at doing this have become stellar at doing this literally through failure. Like this is like, if there's one thing that I can teach you about biology, it is embrace the suck. Embrace the failure. It is all failure all the time. Like, if to be a biologist, you need to get really comfortable with that because your shit is going to die for no reason. Your thing's not going to work for no reason. And you're going to spend 6 months debugging why the hell did your thing die and or not work. It's the way it goes. And then it works and you open champagne. It's just how it is. So to how to get started, just start. You don't like this. You know, if you needed like encouragement or whatever on your way out, fine. But like, just pick a project, something like the whole point of this stream is to show that you can pick something absolutely ridiculous, like making deer milk out of yeast, and you can just do it. And yeah, it does get to be a very expensive hobby. It's biology. It is extremely expensive. This is why you spend several hours or days designing your thing in advance before you commit to actually buying anything. Like, this is why I was taking my time. This is why I spent an hour and a half looking for the correct plasmid, because if you didn't, you end up burning money. You just hemorrhaging money. So it is always better to take your time, think it through, think through the ways that it can go wrong. Check it with somebody else. Like, just make sure that you like are 100% sure of the thing that you're about to do. Then you make the commitment and actually spend the money, not beforehand. But yeah, very good. So cool. We now have a new plasmid made, ready to go, ovalbumin finished. Now we can start on deer milk. So we're going to go whitetail, deer, beta, casein. Let's see what comes up. Uniprot. So yeah, Uniprot is usually a good start here. I need the name of tail deer. Whoop, deer. I just need the species name. Yeah. Okay. So the way that we're going to do this one, we actually have to do a little bit more. Going to have to do a little bit more actual searching for this, because Uniprot probably has it, but finding it's going to be hard. So, to just answer Timothy's question, yes, there is a community of people who you can check this with. There's a few different groups on Facebook. If you look for genetic engineering or biohacking, you'll find them. Just go, be cordial, be nice, be polite. Don't be a obnoxious dickhead the whole time, and people are pretty good about helping you out. So yeah, that's good. Okay, so because the deer proteins are not coming up immediately, we're gonna have to do something a little bit different. So we're gonna be using a tool called NCBI Blast. So Blast is basically a search algorithm where we can feed in some bits of information, and it should spit out the information that we want in theory. So first things first, we're going to set organism to white-tailed deer. Now we need the protein that we want. So in this case, we're just going to look up beta casein on Uniprot. Cool. Get beta casein, CSN2 from Le Momo cow. And for those who are just tuning in and wondering, why am I talking about deer milk? Because the companies that are making milk using yeast have attempted to patent every other frickin' animal like the bastards that they are. So we're choosing the one animal that they didn't list. And for those just tuning in, they included woolly mammoth on their list because they're bastards. So we're using deer because for whatever stupid reason they didn't use deer. For those who I just kind of glazed over what I just did. I looked up the cow beta casein. I grabbed the sequence of amino acids. Now I'm going over to the blast window, and I'm just going to paste that in. And I've set the organism to white-tailed deer. And now I just click blast, and now we kind of wait, and it should pop up almost immediately. I tried this before. Yeah, they forgot platypus, but you wouldn't want to drink platypus milk. It's weird. It's like real weird. Like they don't have whatchamacallit? They don't have teats. They like sweat milk, and it's red. It's weird. You don't want platypus milk. 0 of 10 would not suggest. Cool. So yeah, blast worked. Here we go. So this is now limited so it's limited its search by species, and sure enough, hopefully you can see this. It has now, we put in the cow beta casein and it just spit out deer beta casein. And so we can actually look at the slight differences, and you'll notice that so the query is here and the subject is here. And you'll notice that there is some distinct differences. There's like these little gaps where, so this is sort of the thing. Like the caseins should give you approximately, should give you approximately the same texture. Like it should have most of the same properties. Like the changes are so small. So now we should be able to grab the sequence ID, which should bring us to here. And it should give us, yeah, here we go. So this is now giving us the deer protein thing. So we're going to grab the fastest sequence like we did with Uniprot. There we go. And now we can just grab this. And now we can go back to Genome Compiler and we can stick it in the correct hole. And we're just going to make sure we have the minus strand highlighted because for whatever stupid reason it keeps turning that off. And we give it a little pasty paste. We're going to do codon table. Just this, we're doing exactly the same thing that we did with the ovalbumin. So we're using the same codon table, same yeast codon table as we're copying amino acids. So it's got to translate back to DNA. We're going to click OK. Yeah, I don't know if the dangly bits are actually there. That doesn't look right. Here, I'm just getting readjusted. Right. Yeah, I don't think this restriction site is actually there. I think this is fake. Or it just hasn't updated or something. So, I have to deal with that after. To Adam asking about why not human milk, because human milk has been patented. The whole point of using deer milk is it's not patented. Come on, do the thing. There we go. That's not actually more comfortable. No, it's definitely copying the site where the restriction site is. I think it's just a glitch with the thing. I'm gonna make sure that I give this a different color because this is gonna get really confusing really quickly because we've got to stick like 4 proteins in here. So we're gonna do pink because why not? Okie dokie. So now we go back to sequence. Yeah. So if I wanted to make sure that that restriction site is actually there, which I don't think it is. What did I cut out? This is why we don't do destructive edits, because it means I can just open this up really quick and we can stick back the people who patented those types of milk. I have this open. This is the patent for those just tuning in. This is the patent number. Feel free to read it. The company was Moofree. Now they're called Perfect Day, I think. Yeah, so fuck them. And not actually. It's a cool company. They do cool things. They just did a really dickish thing by attempting to patent every animal they could think of. Yeah, see, this is how I know that the restriction site did not actually get copied properly. So TCGAC, whereas this is totally different. So what I'm going to do is I'm actually going to add in those letters. Now, ideally, what would be really good. Felix Marker, what prerequisite question? Yeah, so just to make sure that I'm not. So what would normally be like the way I would normally do this is I'd have a document open, just like taking notes on the changes that I made and this kind of shit. But I'm just not feeling it right now. So I'm gonna go ahead. I'm gonna copy this, and we're gonna just stick this back in at the front here. Actually, you know what? I'm not gonna do this yet. What just happened? No. Let's see. Undo. Okay, yeah. I'm gonna not do that. I'm gonna fix this restriction site at the end. After I got one more thing to stick in here before I can do that. And then I can fix it after. So again, we're gonna do the same thing because we want excretion. Oh shit. No. Select the whole thing, please. There we go. And before I do anything, I'm going to go ahead and make my copy. Scene and secretion. And put in milk and eggs. And yeah, I mean, so one of the nice things with this is it would be inherently lactose free. Like, unless you added lactose to it, like this would be milk that is lactose free. It's probably not going to taste like milk. It's probably not going to look like milk, but it's the proteins from milk. So you could probably make cheese. Like, actually making it into milk means you'd have to add, I think it's like triglycerbutyric acid or something. There's like, I think it's called tributrin. It's one of the flavoring compounds. You'd have to add a sweetener. You'd have to add plant-based fats or just fats. It doesn't really matter. If you're going plant-based, then stick with plant-based. But whatever. Okay, cool. So I've already got the secretion tag. We're gonna just go ahead, do a little pasty paste. Yes, all this software is free. It's called Genome Compiler. You can just, translation table. Standard code. Keep original sequence. Yeah, all the software is free. It's just called Genome Compiler. You can just make an account. It's totally free. It's one of the reasons I love it. So just for the sake of this, I'm going to just give this a little color change. So just for consistency, we're going to go with, puke green as the excretion color. So because we're going to actually be pasting this in a bunch of different times. So yeah, this should just do the thing. Need a little bit of a stretch. Insects don't make milk. That's a mammalian thing. They don't make milk. What else we got? Yes, this live stream will be available later. I'm leaving it up. I'm just kind of like taking a little break to just answer some questions because rice milk is not milk. It's rice juice. Lewis Black has a really great bit, and I realize Lewis Black is not a comedian that many people know anymore. But he has a really great bit about like soy milk is not milk, it's soy juice. You can't say soy juice because as soon as you say soy juice, you just start to gag a little. Okay. Yeah, that's enough questions for now. Cool. So, I mean, right now we have a plasmid that would make a deer milk protein and would secrete it in yeast. Cool. Now, the thing is, as we talked about way back in the beginning, before I got sidetracked for an hour trying to find a fucking plasmid, one of the things is it's not one protein that makes milk; it's many. Specifically, there's about 4 of them. So we are going to need to add more in. So we've stuck in this pink thing. But now what we're going to do is we're going to do something a little interesting. So there's this really cool piece of DNA called a P2A spacer, where basically what it does is once this bit of DNA is transcribed into RNA, as the ribosome is copying, or not copying, but as it's reading the RNA and turning it into protein, it'll hit this P2A section, and basically it kind of skips. So it stops making protein for a second, like ejects the protein that it's already made, skips a few letters, and then starts reading again after the P2A. So this is really helpful. So you can use one promoter, so it only needs to make one big piece of RNA, but you can make multiple separate proteins from the same single piece of RNA, and this is really, really helpful. Okay, quick question to answer. Arjun asks, what do I need as prerequisite knowledge to get into genetic engineering? As much bio knowledge as you can cram into your head. Bio is an extremely, extremely deep field in the sense that there's just so much. So it's one of those, like, pick a project and just work through it. It's the only way that you're going to get through this. Like, I'd suggest picking up the manga guide to molecular biology. It's a great book for giving you the basics. You know, read a bio textbook, read a molecular biology textbook, learn as much biochem as you can cram into your face, and then just pick a project and work through it. It's the only way you're going to get through anything. But it's not that bad. Like, once you start, it's not so bad. So anyways, I was talking about P2A. So we are going to go ahead and grab P2A sequence. AddGene, so they're called self-cleaving peptides. Let's see if we've got one handy. Yeah, there we go. That's what I like to see. GSG. Boo. Yeah, so there's basically 2 ways to get multiple proteins out of a single piece of RNA. One of them is an IRES sequence, which is basically it's almost like a promoter. It's the ribosome will just grab onto it directly, whereas P2A, it just skips the a little bit of thing and then keeps going. Or is it self-cleaving? I can't remember. It's yeah, thanks for the reminder about the dangly bits on the egg white. I'll go back and fix that. Just remind me once I'm done this, and then I'll get in there. Yeah, so I was okay. So the 2A peptides are mislabeled, and it says it in this paragraph. Basically, the self-cleaving is how they're referred to, but they don't actually cleave. It's the ribosome sort of skips. It like stutters. It's like, think of a record skipping. Okay. Just want to get rid of this. Oh, come on. Really? There's spaces? Damn it, AddGene, you had one job. So I'm just cleaning this up so that I can paste this in to the thing really quickly and easily. All right. That'll do. Copy. Cool. So now I've got my 2A peptide copied. So this is going to let us make multiple proteins from one promoter. This is the whole point of doing this. So now I can go to Hopefully right here, if Genome Compiler will fucking cooperate. Come on, load, you bastard. There we go. Pasty paste. We're gonna use codon usage. Yeasts. Okay. That's highlighted. We're gonna annotate this. It's always important to annotate. Any new phage projects? No, I don't fuck with phages. As soon as you've got phages in your lab, it ruins your ability to do genetic engineering. I don't fuck with phages. Okay, so in the same way that we've got our excretion tag being puke green, I'm going to give this another color. What would I even label this as? I mean, it is technically a coding sequence. It's just weird. I'm going to make it, I don't know, what's a fun color? As to how will I regulate the levels, I won't. It's just I want basically flat levels of all of them. This probably isn't ideal, but I mean, I don't care. We're just kind of going with it. I mean, it's something to work from. Like once this is synthesized, should you have a problem where you actually need to mess with the levels of individual things, you can clone them out, you can mess with it. But like, I need something as a baseline. So we're just going to go with this. It's probably not going to be perfect, but we're also making deer milk. So, I mean, I've never made deer milk cheese, nor do I know anyone who's made deer milk cheese. So it's going to be weird no matter where we start. Yes, the quality is only 480p because I'm a turnip and I don't know how to fix that. That'll be fixed for the next stream. Why do phages screw up your lab? Because phages are viruses, and so they get on everything. And if you have a phage that can kill E. coli, and you do genetic engineering that requires E. coli to work, if they ever get into your culture just dies. No reason. So I do not mess with phages. They're the worst. Okay, I think that's... okay, I'm just gonna have a little... Chikaroo. Phone has been exploding, but that's okay. Need to go into settings, click on the output tab, edit the output resolution, video tab, scale. Okay, wait. Actually, I just got some. I'm gonna take a little breaky poo to fix the Streamlabs thing. So just give me a second. Cool. So I'm going to just mess with Streamlabs for a second, see if I can fix the output resolution. Okay, go into settings. Go to the output tab. Oh, okay. I can't mess with it while the stream is running. Okay, well, I'll have to fix it for next time. Sorry about that. I know this stream is not in, beautiful HD quality. I apologize. It is the way that it is. Like I said, I'll fix it for next time. So, but yeah, next time we'll do full HD. It should be fine. This is really frustrating. I'm sorry about that, but it is the way that it is. Cool. So, back to the task at hand. We now have one of probably 4 proteins in the thing. So we've got our pink, which is the beta casein from deer. We've got our P2A, which is it's a little skip. and that should make things, keep on keeping on. And now we need to do the next protein. So we need kappa casein. From Uniprot. Just as soon as my computer stops lagging. Yeah, like I said, I'll fix it for next time. Sorry about the resolution, but we're already like 2 hours in, so. Also, the nice thing is now that I have this plasmid already like loaded into Genome Compiler, the next times that we go to do this should be a lot faster. Like a lot faster. That really ate like an hour of the time, and that I realized was kind of boring, but it is the way it is. Okay. I'm glad that you guys are finding more mammals that we could have used. I appreciate that. So, this is the thing with patents, right? Like, someone's trying to be a dick and patent every single one of the this is a fragment. Why is this a fragment? I don't want a fragment with the whole thing. That's stupid. There we go. So yeah, this is the thing. Like, somebody tries to patent all the animals, they forget that there's a lot of animals. Again, this is a fragment. What the fuck? Okay, this is fun. Just give me the whole frickin' protein, please. There we go. That's got an M on the front. That'll do. Complete. Okay. So this is actually from some other species. I don't actually recognize what this species is. So I'm going to go back because I want to check. Oh, here we go. Yeah, so this fucking water buffalo. So I was just trying to find the kappa casein protein. And for whatever reason, the cow version was only fragments. Like, it wasn't the whole thing. So I had to just check for a different species. And the one that had the complete protein is actually water buffalo, of all things. So we're going to use the water buffalo kappa casein, and then we're going to look for... I closed that, didn't I? Yeah, I did. Need the species name again. There we go. I mean, we could use water buffalo because I don't actually think that was in the patent, but for consistency, I'm going to keep using water buffalo or deer. Gnu milk. I can't pronounce your name. It's in Russian, so sorry. But thank you. Yeah, whale milk. Fucking whale milk is in the patent. There we go. We've got max score 100%. Perfect. Perfect. Okay, cool. So we have found the kappa casein from deer, because it's that kind of day. Give it a little copy. Pop over to Genome. Come on. Drop bear. Yeah, koala milk would, I think, be peak weird. What made me choose Genome Compiler over other software? It's free. Also, it's just really easy to use. Like, it took a little bit of getting used to, because I mean it's dealing with biocode, but like, it's so nice and easy to use. It really is just an absolute pleasure, comparatively. Cancel. I did not do my non-destructive edits. Whoopsies. Come on, save as. With and scene and secretion. Okie dokie. Oh, milk and eggs. So yeah, I'm gonna actually just skip a step rather than saving after every single paste, just because it's annoying. Yeah, mammoth milk has been patented because they're bastards. Okay, what else we got? I'm trying to follow along the chat, and I'm also trying to just get this done because this has already been like 2.5 hours. But yeah. Okay, so I'm gonna go ahead. I'm gonna paste in the kappa casein from the deer. We're gonna annotate it so that we get this done. Just as soon as Genome Compiler feels the need to get on with it. Change this to a coding sequence. Call this kappa casein. And we're going to give it a color. I'm thinking, I don't know. No, not red. Lovely shade of purple. I mean, we could make blue alien milk, but I think what maybe we'll do that at the end. Just let's just get this done, and then if we're feeling a make it blue mood, we can do that. Yeah, so I think it's not the genes themselves that are patented. I think the way that they got around the you can't patent nature law is they're patenting the use of those particular proteins in their particular way. However, because we're using a different protein, they've got nothing. So fuck them. Okay, that's been labeled. Back to the sequence. And then at the front of the new protein we just added. Cool. So now we have deer beta casein and deer kappa casein. So we're like halfway there. Cool. Now I actually have to check the patent, and because I think really having those two is like most of the work. One of the other ones is lactobumin. Come on, you bastard. Work. Why are you highlighting protein? So yeah, I'm just scanning through the patent really quickly. Let's see if I can turn this. Yeah, here we go. Okay, so this is the important bit. So the other things that could be included beyond the caseins are lactobumin and lactoglobulin. So we're gonna have to find both of those, and then we can throw them in, and we're done basically. So yeah, let's give those a find. Okay, so yeah, so it's alpha lactobumin. Okay, wow, that's annoying. What is this? I think there's actually 5. What is this? No, that is not helpful. All right, let's find some alpha lactobulmin. Yeah, yeah. The only actually shitty thing about GMOs is the patents. The rest of it is fine. Um, so asking about so I saw a thing saying the cacao genome has been sequenced. Yeah, it has. Chocolate is very complicated. Like it's something like 400 different compounds all coming together that actually give the flavor of chocolate. So somebody suggested chocolate milk cows. It's not possible. I mean, it's not entirely impossible, but it's functionally impossible. Um, so it's just not even worth it. Okay, so oh, shit, I gotta stop kicking my mic. That is really it's really annoying. Um, Patents encourage nothing. Patents encourage being a dick, and sitting on ideas, because then some asshole can, like, you could have a really cool idea, and then some asshole could be like, wow, that's a really cool idea. Let me buy that from you, and then put it in a box and then sit on it for 100 years so that no one's allowed to play with my toys. It's awful. Um, okay, I'm gonna leave that open so I can use that. Let's go back. Oh, too far. Too far. Okay, cool. So I've grabbed the, I don't know, I wasn't really narrating that, but this is the alpha lactobulmin from cow. We're going to do exactly the same thing that we've done 2 other times, which is I'm going to grab the species and I'm going to throw the protein in here. I'm going to tell it to find it from this species. We're going to run blast and we're going to see what comes up. And while it does that thing, I'm going to answer some questions. I mean, patents can last anywhere from 25 to 100 years. Or no, it's copyright that lasts 100 years. It's 25 years, I believe, um, is the length of a patent. But you can also there's ways of extending them. Like you can make slight changes and then basically refile, or you can just file them in new countries, and it's obnoxious. Um, yeah. So ideally, like in Magical Wonderful Funtime Land, like a patent should last 5 years. It should give you just enough time to develop your thing, make like it like get your market, and then fuck off and let everybody else share. Because like right now, like patents are the worst, especially with like biotech patents, because they. Oh wow, that's interesting and weird. Yeah. Yeah, like 20 years is a long time. Like 20 years, the amount the world has changed in 20 years, like it's indistinguishable. Um, so being able to sit on an idea for that long is just inexcusable. Like 5 years, I can understand. It gives you time to get your market, like get in, get out, do the thing. But 20 years is just awful. Um, it takes more than 5 years for a medical biotech product to come to use. Yeah, that's because the medical situation is equally obnoxious. Um, anyway, um, okay, we've got my thing. I'm gonna go over to here. I'm gonna do my save as because I'm not gonna forget to do that this time. And alpha lactobulmin. Um, I like open patents. I like the idea of personally, I'm a big fan of copy left, which is you basically like either buy the patent or just patent as many things as you can in such a way that it forces the patent to remain open. So that way nobody's allowed to be the sole owner of it, and then everybody gets to play with it. Um, How are biotech firms supposed to make any money? By making a good product and selling it. Same as everybody else. You don't need a patent to do that. Like, it just encourages competition, which is actually a good thing, last I checked. Okay, so we're going to grab my P2A again. We're going to go into the end. Also, you're never going to see me cry over, like, a biotech firm being unable to make money, because they're always able to make money. The idea that they're not is ridiculous. Okay, we're going to paste in my P2A again. Good. Stefan, you're making my point for me. If their competition makes it cheaper, that's to our benefit. Fuck them. Like, oh no, they can't make billions of dollars. They can only make several billion. Like, instead of making like 30 billion, they make 10 billion. Oh no, let me go cry in my Ferrari. Like, this is, I don't care. Like, okay, we had the thing. I did the thing. Where did it go? Alpha-lactalbumin. Here we go. Copy this again. Just stick this in here. Yeah, like in theory, patents are great. Like it theoretically allows people to do business. Except the way that business is done these days is indistinguishable from the way that it was intended, and it's just people abuse the system by having huge teams of very expensive lawyers, and so for that they get 0% of my sympathy. Okie dokie. Alpha-lactalbumin. Let's pick another lovely color. I'm gonna go with a blue this time. Feeling kind of blue. Yeah. Also, the number of people I know who, spent years of their lives developing something really cool, and then the person who was funding it going, "Oh yeah, by the way, you didn't notice the fine print in your contract. I own the patent to the work you've just spent 5 years developing. Your choice is now either work for me and do exactly what I say, or fuck off." So, yeah, fuck them. Not into it. I mean, then there's the argument of, yeah, if you're a small company and you make something, a big industry could steal it. Yeah, sure. Except that the big industry would have to retool their entire factory to be able to do that, and they have a million other things that they're doing, and China will just steal it anyway. Like this is the other problem with patents. Patents kind of protect you against people in North America. They don't protect you worth a fuck from people in China. So yeah. What was Lord of Chemistry's question? I don't even see it. Yeah, like the 3D printer industry happened because the patent expired. Like insulin being available happened because the patent expired. Like, eh. Yeah. Also, that's the other thing. Like with whatchamacallit? Like if the argument about like, a patent protecting you from a big company stealing it is not true. A big company has huge amounts of resources and lawyers and such to parse through your patent, find the loophole that you are definitely going to have missed, and then still steal it from you. So like it doesn't actually protect shit. It protects them from like sitting on it. Like there's a patent that the logging industry owns that makes trees, and any plant really, grow twice the size in half the time. Except that's a patent. Like it is the it is the tightest patent I've ever seen. We cannot think of any way to get around this. And so they've literally found a way to like solve huge like if you could make all the plants just grow twice the size in half the time, like that's a much larger amount of carbon that you could sequester. There's all kinds of different things that you can do with that. But they've locked it down and you're not allowed to touch it. So fuck them. This is I don't like patents. They're the worst. Okay, we got beta casein, we got kappa casein, we got alpha lactobutmin. The last one that we're missing, and then we're fucking done, is lactoglobulin. Yeah, lactoglobulin. Beta lacto. Oh, it's lactoglobulin. All right. Uniprot, please and thank you. Oh, this is whey. Okay. Got it. Okay, so the casein is the like chunky, like it's the white parts of the milk, whereas the beta lactoglobulin and the alpha lacto, sorry, the alpha lactobumin are the whey proteins basically. I mean, you can try and just use their ideas, but then you can get the shit suit out of you. This is why it's important to find ways around the patent rather than just straight up stealing the patent, because that's the way the system works. I mean, this is the same in the medical industry. Like someone will come up with some drug, and then basically all the other companies will just start sticking functional groups on it until they hit one that has the same effect, but is just outside of the patent, and then that's how you get your generic or your off brands. And they could have almost an identical effect, but as long as it's within a certain amount of time or a certain amount of stuff, they almost don't need to. Like I can't remember the exact specifics, but it's like, they don't need to run as stringent of clinical trials as long as they can prove it's equivalent. I mean, it's not even conspiracy shit. It's just like some people just buy patents and sit on them. There's many cases of, companies buying patents from smaller companies that have produced a better product, or an improved method, and then they just sit on the patent and continue doing it the shitty way, because they made them an offer they can't refuse of like an enormous amount of money. Then they just sit on the patent and can keep doing it the shitty way just because they can. Okie dokie. So we're going to do another save as. We're going to add... Oh, I forgot the secretion tag. Cancel. I'm going to just do that really quick. Copy. And then we're going to go over to here. Just to recap on BLAST, what BLAST does is you can give it a either DNA sequence or amino acid sequence, and then you can also specify a species, and it'll try and find a sequence in the NCBI database that is as close as physically possible to the thing that you want. So in this case, I'm putting in cow proteins, and I'm telling it to find the equivalent in deer. Okay, cool. So now I've actually got my secretion tag in there. Okay, so just to review, we've got secretion tag, beta casein, P2A, secretion, kappa casein, P2A, secretion, alpha lactobutmin. I'm going to add the P2A after I do this next save. And the last one we're going to do is beta-lactal globulin, and secretion. Milk and eggs. It's very rare that little guys refuse the offer. Somebody offers you many millions of dollars, you usually take it. Like, because you'll probably make more from that sale than you would actually developing it. Like, it would take you 15 years to actually develop it up to the market scale of making the equivalent amount of money. And yeah, sure, in the very long term, you'd probably eventually outpace them, but like it's hard to tool a factory and get it all going. I'm not planning to adjust the concentration levels. This is like proof of concept. This is just get it done, get it to make some weird milk stuff, and then go from there. You wouldn't. Come on, grab it. There we go. You for a prototype like this, you wouldn't try and mess with the levels. You just want to get it made, and then you kind of go from there. So in theory, this should make approximately even levels of each protein, which is not going to be particularly accurate to the way that milk exists, but it would still give you a pretty decent approximation, and it would still probably make some cheese. It just wouldn't be, because it kind of depends what you're going for. Like, I'm just going for make a thing that gets you all the proteins so that you can make cheese. Because the nice thing is the alpha-lactalbumin and the alpha-lactal globulin will stay in solution for the most part. Whereas if you acidify the thing, the caseins drop out, that's what cheese is. Cheese is basically, you take all this mixture of proteins, you acidify it, and the caseins drop out, and the whey proteins stay behind. So in this case, this should give you a reasonable, but I mean, some of the whey proteins do also drop out, just not as much. So because you're getting a mix of all of them, you should end up with something that's a reasonable approximation of milk and would behave approximately the same. It's not going to have the fats. You're going to have to add those separately, because otherwise you'd have to do fat synthesis, and that's just no fun. Okay, beta-lactal globulin, copy, and I think we're done. Okie dokie. So we're going to do codon table. We're just doing the same thing over and over and over again, which is use the yeast codon table, paste the thing, and call it a day. What if magic was real? Magic is real. It's called science. Think about it. I'm putting some things together on a computer, and I pay a company, and they make it, and then yeast make cow milk or deer milk. Sorry. So yeah, I think as these things go, this is as close to magic as you're gonna get. The alchemists of old would be very jealous. What's the difference between cream cheese and yogurt? Yogurt is a fermented product. So the thickness comes both from the proteins crashing out in weird ways and also the stuff that the bacteria produce and like cross-link proteins in weird ways. Whereas cream cheese is it's cheese. You crash out the proteins and then you blend it. So who just thanked me for the answer? Whose question did I answer? Oh, he retracted it. Okay. Yeah, I know. I've got to fix the dangly bits. I'm just labeling this. I can spell. Lactoglobulin. Okay. Holy crap, I think we're done. Oh no, wait, I wanted to change the color. I mean, at this point it doesn't really matter, but I want to anyway, because I'm a nerd and it sounds fun. What's a good color? I'm gonna go bright pink, just because why not? I've been doing like light pastels. Were you a dairy expert this whole time? No, I can use the internet and I can read patents. What else we got? Yeah, if I actually do get the, so this one's going to be expensive. Like the egg thing I can do for about $400. Whereas this one I'm actually gonna check in just a second. But my bet is that it's gonna be very expensive comparatively. Actually, that's not so bad. I mean, it's not nothing, but it's not so bad. Okay, 3580. 3580. That's a lot smaller than I thought it was gonna be. Huh. Interesting. Okay, that's on the low end. Yeah, okay. So to actually have this plasmid made would be in the range of $700 to $900. So yeah, it's a little pricey. It's a little bit outside of the range of something that I am comfortable just throwing money at necessarily, but it's something that I could do a fundraiser for, and then it'd be a lot more comfortable. Even if it doesn't cover the whole cost, if it covered like a chunk of the cost, I would be a lot more comfortable throwing some money at this and having it made. And then yes, any of these plasmids that I have made will go on AddGene. The spider silk will go on AddGene. Also, these files are going to be loaded onto GitHub so that anybody's allowed to download them and play with them and do whatever they want with them. Please don't crash. Okay. Somebody keeps asking, couldn't you use cow protein with an extra bit? I mean, technically. Oh, the concentration level. Okay, cool. Glad I answered that for you. Yeah, I mean, you probably could, but that's actually more difficult than just using a different protein. Also, if you use a protein from a different animal, you have the benefit of saying that you took it from a different animal. And there's literally a live stream of watching me take it from a different animal. So, I've got the evidence to back that up. When will you make another food video? Probably this week. I literally have the ingredients for it right here. I'm doing a thing on harvesting yeast from wild sources or extending the yeast you already have, because apparently people are baking wonderful, wonderful bread all the time now, and they need yeast. How long will the stream be? Well, I'm almost done, so we're on to the wrap up. I'm just kind of answering some questions. Then I'm gonna do a little bit of tidying up on the plasmid because I got to fix some shit at the beginning. Actually, let's go here, and then this should be fixed. Spider silk protein is not easy, actually. And the real properties of the spider silk thing is that's some like esoteric, like people are like, well, what will it have? It'll have the same properties. It's the properties of spider silk come from two things, which is the sequence of amino acids and the length of the thing. The rest is not actually hard. This whole like it needs mechanical forces during, no, it doesn't. It just needs to be large. All the research that I've found is big protein give same properties. It doesn't. The like esoteric it must come out of a spinneret thing is bullshit. It doesn't. Did I not add a secretion tag? Did I miss one? No, I got 4. There's a. The secretion tag comes before the protein, and they're the puke green ones. I've got one on all of them. Yeah, I just gotta fix the dangly bits, and then we're done. Am I a microbiologist too? In the sense that I handle microbes all the time. But I mean, if you're asking me to like figure out like what kind of microbe something is, it's not really. Grow a bunch, do a gene extraction, send it off for sequencing, and have my answer. But other than that, yeah. I mean, I grow a lot of fungi and I grow a lot of different organisms and stuff, but like, I'm mostly I mostly do molecular biology. As to the making a video about the testing of the infamous virus that's going around, I'm literally working on that video. I have all the supplies in my fridge. I'm actually waiting. I'm actually trying to show as many different tests as I can. So I've got the PCR-based one. I've got a friend who's going to be sending me some of their isothermal-based one, and in theory, I've got an antibody test on the way as well. So I'm trying to show it all off, which is why it's taking so long. Are there other molecular gastronomy videos planned? Yes. How do you detect, fix potential copy-paste errors when adding sequences? You be very careful. I don't. That's all I can say. Just be very, very careful. There's a P2A. So the P2A I've added to all of them. The P2A basically, I'll just review this really quick. You know what? I'll wait on that. I'm gonna do a review once I gotta fix the dangly bits, and then I'm gonna do a review of everything that we did on the stream, show off the two things that we made, and then kind of go over them all. I mean, this is so you. I mean, this basically is growing cheese. That's sort of what it's going to be like. It's going to depend on if the proteins crash out or not, which I don't know if they will. They probably will, though. How does the company actually manufacture the plasmid? They use a chemical process to print out each individual DNA base pair in short little 200 base pair sections. Excuse me. And then they put all those sections together into the actual insert that goes into the plasmid. Then they cut open the plasmid and stick the insert in. It's something you never actually want to have to do by hand. It's the reason why you pay companies to do it for you. It's a fucking nightmare to do. What are my educational qualifications? I have none. Do you think it's important? Fuck no. All of the best biologists, or not all of them, but a lot of the best biologists I know have no degree. But in the same breath, I also know a lot of great biologists who do have a degree. It's not the piece of paper that makes you a good biologist. It's your ability to do biology. I realize this has just turned into like question time, but I just kind of want to. We've been doing bio for so long that I kind of needed a break before I go back to it. No, I'm not going to delete the stream. The stream will stay up. How do the secretion tags work? The secretion tags are a fragment of a protein called alpha mating factor, which yeast normally secrete as part of their mating process. The internal machinery of the yeast recognize the little front part of the protein that has the alpha mating factor chunk in it as anything that comes after, like this, and anything that's attached to it needs to be exported because it thinks it's alpha mating factor when it's not. How did you find out what was patented? I looked up the patent, and then I read the patent, and I showed the patent. How did you learn this stuff? The hard way. Are you doing a Western blot or ELISA for the antibody test? No. If I never need to do a Western blot or ELISA in my life, I will be grateful. I hate doing them. I bought one of the little, and I mean, Lord knows if it'll actually show up. I found it on eBay, but I, in theory, managed to get one of the like 15-minute fast tests that you like put a drop of blood on. So I just want to, assuming it shows up, I'll be able to demonstrate it working and then talk about how it actually works. But it might not. I might have to omit it. Where are the bioluminescent trees? Actually, I'm working on it, but that's a story for another day. How's the Neuron project? The Neuron project is progressing. If you have not seen it, go watch Ben's new video on applied science. He's helping with that project. He's making the base plates. He's amazing, and he's making a lot of progress. Go check out his video. Once he's done with that, then I'll make... Well, I mean, he's got to finish that, and then the pandemic needs to fuck off, and then I can make the next part of the video. What else we got? The hard way is you pick a project and you just blunder your way through it until it either works or you give up. Um, but I'm stubborn, so I never give up. Um, what else we got? How did you get this to this point without having to bow to the whims of people who hand out the shiny bits of paper? Large amounts of personal expense and starting a YouTube channel that lets me fund these experiments and fail a lot. Um, I did a couple years of university. I did like 2.5, 3-ish years of actual university studies in biology, but I very quickly got bored of it. It wasn't most of the things that I use on like a data, like it taught me a lot of the like basic like lab handling stuff of like this, but I mean I've always had a lab in my house. Like I've always had the home lab thing. Um, so yeah, I mean it kind of taught me some. I mean, it taught me a bunch of very valuable skills, but it did not teach me the majority of what I know. Most of what I know came from doing it the hard way, picking a project, working through it, failing through it, failing over and over and over and over again until I got good at it. How's the spider beer going? The spider beer is going. It's almost done. I think I accidentally made some silk. So like I took the DNA that's supposed to go into yeast and I put it into bacteria just to make sure that I had a backup. And the bacteria behaved very weird. Like they were very goopy. Like I've grown a lot of E. coli. I've never seen them do this. So yeah, I think it worked. So the thing that's preventing me from making the video is that I've been having issues getting the DNA into the yeast because the yeast species that the DNA was built for is stupid, and slow and just very difficult to transform. So, yeah. So what we're doing is we're basically taking the DNA that we had made, and fuck it, I can just open it. It's right here. It's this guy. So this is since we're taking a detour to talk about spider silk. Wait for it to load. Yeah. So this is the DNA that we had made. It is sitting in my freezer. I have an enormous amount of it. So yeah, the issue is that this is built to work in a species of yeast called Pichia pastoris, which is very difficult to transform. So what we're doing is we're going to basically, and I actually think I have this open somewhere. Where did I put it? Or did we make a new one? Anyways, the point is, but what we're going to be doing, in fact, I might do that later today or tomorrow, is we're going to be cutting out this whole silk chunk, and then sticking it into a bacterial plasmid, so that way we can just have the E. coli produce the silk, so that we have a proof of concept. We have some silk being made, and I can make the damn video, and then I can just take my time and figure out why the hell the yeast isn't working. But we're almost done. Like it's working. I might have made some silk, not sure yet, but like we're getting there. Okay, I'm gonna do one or two more questions, and then I want to finish this up, or at the very least, I want to finish the plasmid before I do any more question times. Favorite sci-fi book? I don't really have a favorite sci-fi book. I do have a lot of sci-fi books I like a lot. I just finished reading Sphere, which was really good. Starfish is another really great book. I don't know. There's a lot of sci-fi books that are great. How do you deal with burnout? Taking breaks. Can you get a job in biology by learning on your own without a degree? Probably no. It's very hard. Even having a degree doesn't, like 75% of the people who get biology degrees don't get a job in biology. There's not a lot of biology jobs. Most of the people who have biology degrees end up as a barista or growing very good weed. But other than that, like it's or you end up as in a pharmaceutical company or you end up doing academia. But like getting a bio job is very hard. This is one of the reasons why I went this weird route where I just like make YouTube videos. The antibody test I'm getting is something that I found on eBay. I don't think it's there. It's not easy to find. It's not easy. Like the listing was removed, and I don't know if it's actually going to show up. If it doesn't, then I'll get my money back. But I got it just to see if it would show up, basically. What else we got? No, the YouTube is my full-time job. Like making videos and doing crazy weird shit is how I pay my bills. So like when people donate or become members or become patrons, that's what lets me do all this stuff. And I mean, I'm extremely grateful for your support and all that kind of stuff. But yeah, I have no other job. This is my job. Doing this is my full-time job, and I spend enormous amounts of time doing it. The lactose thing, I'm actually going to be doing an update video. It was supposed to come out last week, but I had an issue with it, so it'll probably be out next week-ish. So yeah, I'm not talking about it now. I'll talk about it then. The hydroxychloroquine thing is it was shit research, and it was basically shown to not work very well. Okay, fine. I'll finish the plasmid. Yeah. I'm also gonna save because, yeah. Come on. And it's lagging. That's exciting. Okay, yeah. I just gotta copy the dangly bit. And then go back to here. And just stick that in so that I don't lose it. Come on. There we go. Pasty paste. Use the original sequence. Paste. Come on, work. Any day now. There we go. Do I have any plans to make bioluminescent trees? Yes. Well, not trees. Well, I mean, maybe trees, but more plants. We're working on it. We're working with free genes to get the thing printed. It's already designed and ready to go. Will you make this DNA real? Maybe. It's expensive, so not sure yet, but maybe. I'm gonna talk to the synthesis company, and we'll see how much it's gonna cost. And then might ask for some help paying for it, because it is expensive. Okay, we got the dangly bit back into the thing. Cool. I'm gonna save as and just make a copy that is more succinct. Deer milk. Beta casein, kappa casein, alpha lactobutamin. Okay, cool. So we're going to save that in milk and eggs. That way it's just a little more succinct, so I can figure out what the hell I'm looking at. Yeah, I'm just putting the correct letters for the restriction site back at the start of the sequence. I'm actually going to adjust the... I'm going to just throw a color on that just so I don't forget it. That way I'll notice that I did it, and so that when I go to pay for sequencing, I remember to include those letters. Okay, what else we got? Cool, and so I'm going to actually just copy that, and then go over to the Ovalbumin, open that back up, so I can just fix the dangly bits on that one, and then we are basically done. And then I'm going to do an overview of what we've made, and then I'll do a few more minutes of questions, and then I'm going to go make lunch because I'm starving. I think rather than a GoFundMe, I think I'm just going to do like a donation bar underneath the streams, because I'm thinking of doing these streams pretty regularly, especially because, I mean, everybody's locked in. It's something to look forward to. Hopefully. I mean, hopefully y'all are enjoying it. Yeah, that way, like, I'll just say what project we're funding, basically, and we'll just, like, you know, just have a donations bar and just show, like, this is, like, I'll do that once I've checked with the company to see what it's going to cost. And then just, you know, we'll see if everybody's willing to pitch in and do that thing. Yeah, see, this one, since I've closed it, it recognizes that the restriction site is gone. So I'm going to just pay. Oh, shit. That's not good. I forgot to unhighlight that. Cancel. Please don't break my thing. Please don't break my thing. Why, thank you, Jesse. Did it? Okay. Did it? Okay, good. Pasty paste. And that should fix it. That should put the restriction site back. Yeah, I don't know how I'll do the donation thing yet, honestly. I like, I mean, I'll have to figure it out. But what the hell just happened? I might do it so it's like basically like a pre-order, basically. So, you know, it would cover the cost of like making the stab, basically. Because I mean, like, making stabs doesn't cost me very much. So I'll excuse me. We'll figure it out. I have not decided how I want to do that yet, honestly. And I'm gonna make it blue so that I forget. But yeah, I mean, I might do it as like a pre-order kind of thing. That could be cool. Or I might do it as, so I don't know. That's something to figure out, not live on stream. I'll have to talk to some people about it. And I mean, the other thing is I might be able to talk to the Free Genes people and see if, like, you know, maybe I can set up a partnership with them, see if they're willing to make this kind of stuff. Not sure. Don't know. All things of, yeah, all things to consider. But I mean, the nice thing is if we had this, it becomes very, very cool. Like now that these are done, like, this is essentially basically infinite milk and eggs, which is pretty great. So considering like a one-time cost of about $1,000, or, well, no, with both of them, it would be about anywhere from $1,200 to $1,500 for infinite milk and eggs is not so bad. So I'm sure I've got some people who'd be willing to throw in. I'm sure some of y'all would be willing to throw in. We'll figure that out another time. The caffeic acid pathway is patented, but we've made some changes to it so that we can get around the patent. Yeah, maybe, yeah, if Twist is willing to become a sponsor, that'd be fun, but I doubt it. Okay, cool. So we've had some questions about what it is that we're making. So I think this is good. And I mean, they're done. So at this point, I can actually share and talk about the things that we've made. So I'm just going to save really quickly, and then I can do an overview of... Oh, thank you, Cosmic Mutant. So now I can actually do an overview of what we've just spent the last 3 hours doing. Cool. Cool. Yeah. All right, we're good. So we've made 2 plasmids. I can close this one now, and I can close this one now. Okay, so now it's overview time, right? So we set out at the beginning of this stream to make 2 plasmids. One that will make regular baker or brewer's yeast produce the protein from egg white. And the other one is to produce the proteins from milk. So in theory, you could brew a sort of pseudo milk and eggs. Now, and I think this would be really cool because even if you wouldn't want to drink, maybe you probably wouldn't want to drink the milk directly. I mean, you could. It could be a really interesting beer. It'd be like a beer that also is kind of milky, which would be interesting. I see these more as cooking ingredients in a pandemic kind of thing, like or just generally cooking ingredients. So with the egg white, you could just brew infinite egg white and then cook with it, make an omelet, whatever. With the milk, you could make cheese pretty easily, and you could also cook with it and that kind of thing. I don't think it would be a perfect approximation to milk. It wouldn't. You're definitely going to be missing fats and flavorants and sweeteners and all kinds of other stuff, but it would be a very good starting point. And you know, it could make cheese, which would be fun. So let's do a quick overview. So we started with an empty, well, it's like what's called a backbone. So it's a piece of DNA that is got a bunch of different features that we want that can have a piece of DNA added to it to give it the functionality that we want. So the core features here, this thing that says can MX, this will give whatever yeast we put this DNA into the resistance to G418, which is a type of antibiotic. There's a different resistance gene that says AMPR. It's ampicillin resistance. So this way we can put this DNA into bacteria and make many, many copies of it very easily. We've got 2 origin and replication sites, which will allow this to be copied in bacteria. We've got a promoter, which is the sort of start, like it's the go sequence. And then the pink, this bright blue and bright pink is what we added to this plasmid. So the blue is a secretion tag. So it says that anything that's connected to this excrete outside of the yeast, which we want. And then the pink is ovalbumin from chickens. So we've got chicken ovalbumin and a secretion tag. So the yeast, once they've got this, will secrete ovalbumin into the fermenter or media around them. And so essentially you could brew a tank of egg white, which would be very cool. Now, moving over to the other one, which is much, much larger. This is the milk thing. So it's the same backbone plasmid, same features. But then the important bits are this light pink, this light purple, the blue, and the bright pink. So the light pink is beta casein from deer. The reason we chose, and these are all proteins from deer. The reason we chose deer is because it's the only, or not the only, but it's an obvious animal whose proteins have not been patented for this specific use. So we've got beta casein and kappa casein. These are the proteins that give milk its like white appearance. It's the majority of protein that makes up cheese, for example. So it gives it that like milky appearance. The blue and the pink are alpha lactobulmin and beta lactoglobulin, which are the proteins in whey. So it's basically going to be a really protein-rich mix of. So if and they all have the secretion tag on them, so that way your face cam is frozen. Did it? What happened? Did it freeze? Like, can y'all still hear me even if the cam is frozen? That's really weird. Why is that frozen? No, this is weird. Can y'all still hear me even though the camera is frozen? Why is this frozen? What the hell? That's really weird. Okay, as long as the audio is fine, then I'm just going to keep talking through this and we're going to wrap this up. All right, cool. So anyways, so yeah, we've got the different proteins from milk, and in front of each of them, we have a secretion tag. It's the same one that we used for the egg white. And then between them, these little yellow bits are called P2A. It's basically what will happen is this whole skips like a record skipping basically, and will jump to and so it'll stop making protein, release the protein it's already made, and then start making the next one. So this way we get 4 proteins for the price of 1, basically. Okay. Oh, the cam is definitely lagging, but it's kind of working. So yeah, this is frustrating. Sorry about that. Don't know why that happened. I might switch. Yeah, I might switch to OBS. I'm not sure. But yeah, those are the 2 things that we've made. We've made an egg white protein or plasmid, and we've made a milk plasmid. Both of these will be posted to GitHub, and I'll post some links under the stream. Like I'll post some links in the description once I've done that. I'll also post them on Facebook and Twitter and all that kind of stuff. And yeah, so these 2, I'm almost definitely going to make the egg white one. The milk one is a little more expensive, so I may start doing a fundraiser for it. And in theory, you could mix these 2 and make a cheesy egg white yeast omelet, which would be strange, but yeah. So that's kind of it. We've done the thing that we set out to do, and hopefully, we will be able to do more sorts of these. Make a ridiculous thing. So I think this is kind of given an overview of like what these streams are going to be like. Hopefully we won't have an hour of getting stuck at like we had today. But yeah, so moving forward, rather than going into this knowing exactly what I'm going to be doing or knowing approximately what I'll be doing, I'm going to actually just draw an idea out of a hat, so that way I'm totally unprepared, and so you can see like, okay, how do you research these ideas? How do you then take that research, translate it into designing a project? Because throughout this stream we've had like a ton of questions of like, how do you get started? How do you get started? This is how you get started. You pick a random, or I mean something you're passionate about, ideally, idea, and you just work through it. You design the thing, you have it made, you work through the you just work through the process. You like try and get it into the organism, try and make the end product, test for the end product, all this kind of stuff. And so hopefully with these 2 plasmids, like if I have them made, that will turn into its own video. And I'm also hoping that some of these ideas are just going to be ridiculous, and I don't want to make them, but they'll be fun to show the design process. But something like this is legitimately useful. It's something that, like, especially in a pandemic where access to materials can dwindle, I think it's important. I think it's something that'd be very cool to share. I think it would be just very cool to have. Whereas some of them are just kind of ridiculous and they'll just be fun. So yeah, that's kind of that. In terms of how frequently I plan on doing these, I'm thinking maybe a couple of times a week. Like um, definitely at least once a week. I think over the next couple of months, I want to start doing this at least once a week. Just taking some time to just design something ridiculous and then essentially force it open source because it's public knowledge. And it's an excuse to just make really neat, weird plasmids. Um, but I might do it a couple of times a week. I might do it less. I'm not sure. But I will announce all of that kind of stuff on Instagram and Twitter and Facebook and everywhere. It like just be sure to follow me on all of those different platforms. And I'll post the announcements of when it's happening. Um, if you have ideas that you want to add to the hat, head over to either Twitter or Facebook, and just post them as a comment under the posts about the streams, and I'll try and go through them all. And I'm not gonna just like put whatever ideas into the hat. I'm going to give them a quick look over and just see which ones are feasible, so that way I don't pull one out on stream that's like I spend 4 hours and make nothing. I want to make sure that I always pick ones that can be made. Like I want to make sure that at the end of the stream there is always a finished plasmid. I don't want to hit a point where I've made I spend 4 hours and by the end of it's like, nope, sorry, it's actually impossible to make chocolate milk cows or whatever. So yeah, that's kind of my plan. I hope you guys have enjoyed. I'm gonna take like 5 more minutes to just like answer some questions, and then I'm gonna wrap it up because I am quite hungry, and all this talk of milk and eggs, and I'm like I really could use some lunch. So yeah, if you've got any questions, I hope you've enjoyed this stream. If you've got any feedback other than the need to increase the resolution, which I'll have that fixed for next time. If you have any suggestions for how this can be improved, or if you, I don't know, just now's the time. The idea can be complicated. I mean, like, things that seem complicated might not actually be that complicated. Um, so just pitch the idea, and then like I'll go through and see how it is. But I mean, yeah, talking about like making a DNA computer, fucking no, that's way too hard. Something that's like here, I'm gonna pull up my list just to have in the background, just so I can read off a couple of the ones that I've got already, just to kind of give you an idea of the difficulty level that we're talking here. So I've got one that says a silica turtle, so like a turtle with a silica shell. Change the color of avocados, growable balloons, black grass, gluten producing rice, glowing animal. Like, just, it doesn't need to be like that simple, but what will you feed the yeast? Mostly sugar, probably a little bit of malt extract, maybe a little. I've got a big bag of yeast nutrient, which is really just a mix of ammonium phosphate and urea. So basically, like sugar, salt, nitrogen. Give them a little bit of everything. Could I go a bit more in depth on stuff that's not so common knowledge? Yes. I mean, it would sort of depend what the topic is. Fusion power videos planned. Probably not anytime. I mean, eventually, sure, but like not anytime soon. I don't have the space for any kind of reactor. Snail with a hydroxyapatite shell. See, like, leave that on Twitter, because that sounds fun and doable. Is there a way to produce oils? Yes, actually, totally. Like, maybe be more specific, or just leave it at that. But leave these suggestions on Twitter, just so that I have a more permanent resource to check, just because trying to get through this whole comment section over the last 3 hours trying to pick out ideas is going to be not really possible. Capsaicin producing yeast. That wouldn't actually be that hard. Add it on Twitter. That sounds doable. You guys have another like 2 or 3 minutes. If you got questions, now's the time. The reason I'm doing bio streams rather than like a radio stream is because bio is something that I can just do on the computer without leaving the house. Because remember, kids, it's important to not leave the house right now. But yeah, I don't know. I mean, maybe if there's a radio thing I can do down the line, we'll see. But bio, like I like this format. I want to keep doing this thing. I think it's something that I can actually do that's valuable and new. What yeast will you transform it into? Saccharomyces cerevisiae. How will you get rid of the antibiotic? Well, so I don't know if this plasmid is integrating. I don't actually think it is. But you could modify the plasmid to have an integration site. So it would stick this whole gigantic protein complex thing into the actual yeast. So that way it becomes permanent. And so you can just grow the yeast normally without having to include the antibiotic. What am I going to have for lunch? A sandwich with some bread that I baked. For transformation methods, I tend to use heat shock. I don't have an electroporator, and heat shock is easy. It just needs hot and then cold. The news on the Human Neuron Project can be found on Ben, Applied Sciences channel. He just made an update. Go check that out. It's a dope-ass video. How will you get it into the yeast? Heat shock. Are you going to remove the tag after protein synthesis? No, it's stuck on. Menthol yeast. That's actually not that hard. I mean, it's not easy, but it's not that hard. And leave that one on Twitter. That one's fun. The hydroxychloroquine, there was a small study that tried to replicate their findings, and they didn't find that it did shit. What else we got? I mean, there'll be more trials. We'll find out. I don't know. How difficult is it to do plant genetics? Not that hard, but it's tricky. News about the Opal project. The Opal project is basically done. I mean, it works. Check out Instagram. I posted a picture of a piece that I made. Really, I just need to run it more times, and that means access to a chem lab, which means hanging out with Nile Red. And at the moment, we're not supposed to be socializing, so I can't really do that. But I've made Opal, it works. So yeah, the video will come as soon as I can go hang out with him and do another run, just so I can get another color. Because right now the one I made is red. Alright, it's been 5 minutes. I'm gonna wrap it up. Thanks everyone for stopping by. I think this was a lot of fun. I look forward to doing this again. I might do another one before the end of the week, but I might be editing, so we'll see. If not, I'll definitely do another one next week, and the week after that, and the week after that. This is going to be a weekly thing. I hope you guys have enjoyed. I hope for those of you who've actually stuck with it through this whole thing, I mean, thanks for sticking around. So yeah, I hope you guys have enjoyed. I hope you'll tune in for the next one. And yeah, have a good day. Stay safe, stay healthy, and I will see you next time.